Professional Fisheries Graduates Forum of India, and Sri Giriyappa Gowda Memorial Award for Best Researcher 2008, Alumni Association US by Alumni Association US Bangalore, Certificate of Merit for Research Activities 2003 by US Bangalore, and appreciation as Best Lecturer by the Farmers of Kundapur and BFD 2008, Certificate of Merit of Research Activities 2010 by KVFS University, Bidar, Certificate of Merit of Research Activities 2011 by KVFSU, Professor KB Ramakrishna Rao Memorial Oration Award 2011 by Sri JCBM College, Sringeri, Certificate of Merit of Research Activity 2012 by KVFSU, and Dr. has almost 26 has served as member Mumbai and OUAT Bhubaneswar, Chairman ICR. He has served as principal investigator on monoclonal antibodies with the 3DBT project, and he has operated NATP, NAIP, ICR, NBFGR projects. He has 40 international and 49 national research articles in scientific journal. He has transferred technology on monoclonal antibodies based on flow-through immunodot kits for detection of EUS fungus and Acromonas hydrophila and Vibrio of shrimp and oxytetracycline and sulfur dimethoxide. He has established a disease diagnostic center funded by DFID UK GOA. He has visited many countries: Canada, United States, UK, Norway, Netherlands, Portugal, Kuwait, Bangladesh, Singapore, Thailand, Philippines, Vietnam, and Sri Lanka, etc. Welcome you, sir.
ನೀವೇ ಮೂವ್ ಮಾಡ್ತೀರಾ ನಾನು ಹೇಳಿದ್ರೆ? ಅದು ಕನ್ಫ್ಯೂಷನ್. Thank you for the heavy dose of introduction. ನೀವೇ ಮೂವ್ ಮಾಡಿ, ನಾನು ಹೇಳ್ತೇನೆ. Very good morning, ladies and gentlemen. I'll be presenting, I'm retired now. I'll be presenting 30 years of our work on diagnostic vaccines in aquaculture health management at College of Fisheries, Bangalore. First of all, thanks for the organizers for inviting me to the parent university as a part of this university. And much of my work, 2/3 of my work is from this university. Then we carved out to a new veterinary and animal science fisheries university at Bidar. First slide, please. With a brief introduction to aquaculture, because most of you are plant science experts here, students here. Fisheries is a growing field, producing about 13 million tons of fish world, providing employment, export, 60,000 crores. I think it is the second agriculture product in India, after Basmati, if I am right. Okay. Yeah, I'll stand here. Okay, but you have to observe one thing. No more it's capture fishes. Most of you know Canada, fishes and then Galakadashtana. No, it was mechanized fisheries, kept on catching fish, and we have reached a plateau world over, including India. Now it is scope for aquaculture, which is growing at 6 to 8% every year, including India and the world. Okay, the scenario has changed. 60% of the fish produced here today in India is from aquaculture. Okay, aquaculture in fresh, brackish, marine water. Latest in non-conventional, your agriculture saline soils, which are less productive, abandoned like fisheries is coming up in a big way. Next one, please. Next slide, please. Can you? Yeah. Aquaculture is growing in a big way. Aquaculture is intensive aquaculture. And if you go to Andhra, Tamil Nadu, it's a model to the whole of world, it looks, aquaculture activity. We were taught aquaculture activity in a 1 acre pond. Now it's all 50 acres, 100 acres pond. Both fish and shrimp. And intensive aquaculture, movement of live aquatic animals uh, across the national, international borders. So also their products. So a lot of disease problem. And rough estimate is we are losing about 1,500 crores because of the disease. And that's why we consider diagnostic and vaccines are very, very important. And in diagnostic, we focused on farmer level detection kits because that's the one which make a lot of impact. A simple one, farmer can do it, sensitive, low cost. So many farmers can use it, okay? Because it's a simple technology. And we made use of the monoclonal antibody because this is the one which can work well at the room temperature, unlike the DNA probes. And they are very specific, uniform, large quantity, and immortal. I am mortal, my clones are immortal. Next one, please. This is the standard hybridoma technology, which all of you know. And of late, it is replaced with the RPAs, recombinant phage antibody system, a library of monoclonals. One need not use the animals, can go to that, but that is with a patent. This is the good old Nobel Prize won technology. 1975, people got Nobel Prize for this. It's very good in diagnosis, treatment, vaccine development. Okay. Next one, please. We produce monoclonal antibodies to white spot virus of shrimp. This is the one which is attracting the world scenario. White spot virus of shrimp. We also produce monoclonal antibody to WMD virus of freshwater prawn. That first one is the marine prawn. And the third one is Aphanemesis invadens. It is a fungus, very rare, in aquatic system, causing deadly disease in reservoirs and culture system. Tons and tons of fishes died from 80 to 90. Now almost all the wild stocks of fish have vanished because of this fungus, deadly fungus. It came from Australia. Now it is in America through aquarium fish. Okay, and we have produced monoclonals to Vibrio, Aromonas hydrophila. These are all the pathogens in fresh and brackish water systems. And we also produce monoclonal antibodies to immunoglobulin M of fish. That's unique rather. What we have is IgM in mammals, but in fish, it is a different type. And it's only the one type, IgM-like. It's not even IgM. Okay. And also, we produce a specific monoclonals to a specific protein of marine shrimp. I will explain the significance later. And two antibiotics, oxytetracycline and sulfadimethoxin, because these are the one highly misused in aquaculture system. As Indians, we are taken for granted. We eat a lot of antibiotics in milk, poultry, fish of late. But being an export commodity, several containers were pushed back to India because we don't want your antibiotic, stuff. That's why we made a farmer level kit for these two antibiotics. Next one, please. We made use of the monoclonal antibodies. We use this flow-through technology for developing kits, farmer level kits. It's very simple. Nitrocellulose membrane of appropriate micropore size, upon which you add the antigen, antibody, it flows down, gets focused because it flows down, flow-through technology. And you can develop a cassette like this. I don't know from a distance you can see this. You can, small cassette, reusable cassettes. Atop there is a nitrocellulose membrane. Okay, next one, please. This is the first test kit we developed for white spot virus of shrimp. That's why I explained this. First, later on, it's a duplication of two other pathogens. We named it Rapid Out Kit. Just it is provided with the 5 solutions, and the protocol is a farmer just macerate the suspected animal, fish or shrimp, put it on nitrocellulose of this cassette, go on adding those reagents, 1, 2, 3, 4, 5, continuously. At the field level, no equipment required. And he gets a dot, if it is positive, at the end of 10 minutes. It's very, very simple. Each test costs about 40, 50 rupees. Okay, purple dot, easy to read. Now it's fine. Oh, two or two cars. Okay. So I have to stand here firm. Correct. Okay, this is the model of the test kit we developed from a distance anyway. And later we came up with a brochure because it's commercialized with all the details in it. This is against the lateral flow. It's not mine. Someone's for the same virus. It's a lateral flow. The one I showed is the flow through. This is very simple, like you can use and throw, but sensitivity is less. I think what we use for corona is this, right? Okay, next one, please. This is the deadly white spot virus of shrimp. It's a deadly virus. If it comes to the farm, it can make a farmer hero or a zero in 2-3 days. Just 2-3 individuals dying first day, second day, third day. If it drags, nothing in it. Okay, so this virus, as such, it is the first one among the aquaculture health problem, causing a lot of damage. It's worldwide, except in cold countries. See the unusual status like coronavirus, large host and age range, prevalence, culture system, water, carriers, wild root from deep sea. Even mother is infected because the infected young ones are let out in the brackish water, and the shrimp come from the deep sea, get infected, go back to sea, and they are collected as mother, and no more a free mother. It's all infected with the virus. 60 to 70% of the mother deep sea are this virus, having one. Next one, please. It's a virus, no chemotherapy, vaccine, because there is no memory in the crustaceans. To develop a vaccine, you should have the memory and the specificity. It's lacking in crustaceans. So avoidance is the best strategy. Intensive screening of the brood, seed, adults, and carriers is the only solution. We have alternatively PCR. PCR is a wonderful technology. I am working on both PCR and monoclonal antibodies. I know both. People are somehow moved to PCR. Everyone is fond of PCR. But I always say, if you have 100 farmers, how many farmers can use PCR? You have to visit a lab, pay money, it takes time. I know it's a wonderful technology. Out of 100, maybe 10 farmers. Suppose you have a simple field level test, which is 100 to 1,000 times sensitive than the first PCR, not the second step PCR, first PCR. That's good enough for field screening. Out of 100 farmers, 90 farmers will use it. Then just imagine the overall impact of the health management. Okay, that's why we focus on field level test. Okay, next one, please. Monoclonal, we made monoclonals to this virus, and this is a Western blot. To show the specificity, Western blot is the best technology. Next one, please. This is the kit we came up with for the white spot virus, which is commercialized now, monoclonal antibody based kit. Okay, it is given to Virbac Animal Health. It's a French company, multinational. Okay. Okay. Next one, please. And we always compare the performance of the test with PCR. 1-to-1 sample. It's just I have shown 5 samples, I think 5 or 6. You can see it is 100 to 1,000 times sensitive than the first PCR. Good enough for field screening. Okay. Next one, please. Yeah, this is what I explained there. This same, I explained the dilution. Next one, please. And also we looked at the performance in the field. How many PCR can detect, how many this can detect. So it's very good, 80%. Okay, up to 80%, which is good. Second step, it cannot be compared to second step PCR. I'm repeating. Okay, first step PCR is good enough for field level screening. Next one, please. Now, where and when rapid out is useful? As I said, mother screening in the wild. Each shrimp mother is very expensive. 25,000 in some season. A mother of this size, 200 to 300 grams, 25,000 rupees in the catching, harbor. And you can't say him that I will go to PCR and come back to you after testing. No, he will not wait for you. So it is very ideal for screening at the brood collection center, young ones by testing by hatcheries, and some hatcheries are duping the farmers with PCR. So farmers can test now this seed. And this is the problem with India. Like I keep telling, PCR is fine. Any sample, a suspected sample, screening is, there is the statistical model, the formula. How many, if you are screening 100, how many samples have to be tested by PCR? If you are having 1,000, how many? If you are having billion, how many? This we are violating in PCR because PCR is costly. Billion also 10 samples, 100 also one sample, no. But with a simple test, you can meet that standard, okay? Okay, PL test by farmers, growth forms, routine test by farmers. As I told you, if it comes to hit a farm, a farmer can be a 0 in 3 days. Okay, suspected mortality, lot of suspected mortality like corona, we have fever of different types, common cold, corona, whatever it is, but specific diagnosis is very important. And white spot is funny in the sense, several agents cause the same clinical signs. So farmer get confused what to do, where to harvest. Emergency harvest is the best medicine for this virus. Farmers go for emergency harvest after say 2 months if it strikes. So this is very good for emergency harvest decision. In a way, it is empowering the farmers. He need not depend on somebody. The next one, please. I'm moving to the next pathogen. It's a deadly disease of the fish. I said between 80 and 90, it struck India. Mass mortality in reservoir, ugly to eat fish. Have you seen a rotten fish moving around in water? Such a deadly fish, and it's caused by a fungus. We developed a monoclonal antibody to this fungus. Aphanemesis invadens. They have named it very well, invadens. It goes through the bone. I have seen in the transcription. It's such a hard fungus. Okay, next one, please. These are the monoclonals, the western blot of the monoclonals we did for this fungus. Fungus is difficult to work with because it's not that immunogenic. Okay, but we could get some monoclonals. Next one, please. These are the monoclonals against that fungus. Next one. We developed a kit for this pathogen as well. Similar kit, a flow-through kit. Next one. See, the result can be read very easily. There's no confusion. And PCR, often we get into confusion like. And we compared with the sensitivity, again here, a pathogen, 100 to 1,000 times sensitive than the first PCR. Next one. Another pathogen, a quiet killer in calf farms. It causes dropsy and body ulcers. I'm sorry, I couldn't add the photograph here. And also septicemia, it's a common problem. It's a ubiquitous pathogen in calf farms. We produce monoclonal antibody to this. Next one, please. And developed a rapidot for this pathogen. Next one. And we compared the sensitivity with PCR. Okay, again, same, 100 to 1,000 times sensitive than the first PCR. Next one, please. And this is another pathogen in brackish water, salt water. Vibrio is a pathogen. We developed monoclonals. I have showed just one. We developed a panel of monoclonal antibodies, about 5 to 6. I just showed one here. Okay, next one, please. I developed a similar rapidot for this pathogen. Okay, next one. Compared the sensitivity with the PCR. This is the gold standard, like we always compared with PCR. Next one. Monoclonal antibody to antibiotics. This is a Herculean task because the antibiotic molecules are small and they are not immunogenic. It has to be tagged with either BSA or ovalbumin, make it bigger, immunogenic, then only go for hybridoma. Next one, please. We did that. Okay, this is the whole procedure. I don't want to explain it. So we just all you see is antibiotic is conjugated with BSA or ovalbumin to make it bigger. Next one, please. Next one. Can you move? We made the hybridoma for this antibody. Okay, standard protocol. Next one, please. And we had to check the cross-reactivity with other antibiotics used in, because oxytetracycline and sulfadimethoxam are the only two permitted in aquaculture. Permitted, there are so many other in this guise. Okay, so also in animal husbandry. Other day I was in Delhi for a workshop on aquaculture drugs, innovators from drug controller, so many. The way antibiotics is misused in India, I'm not talking about used, misused. You don't feel like eating eggs or drinking milk or whatever, including fish, latest one. Okay, so we looked at the cross reactivity and only the two, it cross reacts with oxytetracycline and other AP, OTC, it's a residue, and with others it don't. Yes, next one, please. Here, with the pathogen, I said it's 100 to 1,000 times sensitive than the first PCR. Here, sensitivity has to be compared with the LCMS, liquid chromatography, mass spectroscopy, because that's the one antibiotic, because most of our aqua products go to America, Japan, and Europe. They are very strict. Several containers have been sent back from India. So government of India is very much worried about this. So we have to compare the sensitivity. We further increase our good old Rapidot using silver staining, a further one additional step. With that, we could increase the sensitivity 30 times further. Now here, I am not comparing with PCR, not even LCMS. LCMS is wonderful. But WHO has the MRL, maximum residue level of antibiotics in poultry, fish, whatnot. Okay? This test can detect much, much, much below that MRL. Don't you think it's good enough for field? Okay? How many government of India established 5, 6 LCMS labs all over India? Some worked, some didn't work for, not the machine I'm talking about, administrative problem. So this can go well. Okay, next one, please. For the other antibiotic, sulfur dimethoxin, a similar process. Next one, please. Yeah. And these are the monoclonals we produce for the second antibiotics. Next one, please. Look at the cross-reactivity with the other sulfur drugs and the antibiotics. Only reacts with the two, sulfur dimethoxin and its subfamily. Next one, please. Here again, with the silver staining, 60 times improved in sensitivity. Okay, that's very good. Okay, next one. So overall, what are the important feature of Rapidot is, it is simple to the farmer level. After developing this kit, I have traveled all along the coast in India, explaining to the farmers in some schools, some laboratories, sometime under the tree. It's very, very simple. Okay. Hardly including sample preparation, 15 minutes, and each sample cost about 30, 40 rupees, maybe close to $1 if you commercialize. 100 times sensitive than first PCR, scope for large number of screening, and a high impact on health management, low cost, and shelf life. It has to be kept in fridge. Okay. Okay, next one, please. Now, this is the one I'm talking about. To immunoglobulin of fish, it is something unique. It's a tetramer. And for epidemiological studies, diagnosis, vaccine development, these are quite handy. And each fish is different. Like there are several families of fish. It's not like in human beings, standard IgG, IgM, and So we developed the panel of monoclonal antibodies to IgM of Katla and Rohu. These are the two fish you see in the market. Oh, I'm just finished. We developed the immunodots for detection. I will take your time because you are delayed a bit. Detection of also I have come up with a specific monoclonals for shrimp. Shrimp artificial meat is there, and so many priced meat is there, but in the market, they dupe the people. So you can, using this monoclonal, you can see which shrimp it is. Yeah, the next one, please. And these are the monoclonals to immunoglobulin of Katla. Next one, please. Rohu, the other fish. Okay, next one, please. And this is that shrimp monoclonal antibody to specific protein of shrimp. Next one, please. And we stretch this work to develop drugs and therapy for virus. Okay, by looking at the neutralizing antibodies, wherein how they tuck in. Most of the time, people talk about antibody titers very high, particularly during corona time. What sort of antibody? Is it a total antibody? You are just binding antibody is no good. It should be a neutralizing antibody, whichever virus it is. Okay. And later using the same neutralizing antibody to develop drug against this virus. Next one, please. We looked at the neutralizing antibodies, sort out. Some are neutralizing. Yeah. Next one. This is how much protection you are getting. This is in vivo after injecting the virus and the antibody to the shrimp. Okay, because there is no cell culture in shrimp. You can't do that in vitro. Next one. And using bioinformatics, we could get some data where the antigen binding to the antibody. Okay, so that you can develop a drug molecule which can tuck to the virus and stops its replication. Yes, next one. I'm not good at bioinformatics. These are the work done by my students who are very good in bioinformatics. How the virus protein tucks with the antibody, IgG, CDR, complementary determining region. Okay, next one. Similar work where the amino acids tucks with these amino acids of the antigen. Next one. Next one. And with that, at that stage, I stopped the work. I left it to my juniors. I retired 6 years back. Okay. This is the second part, oral vaccine we developed for fish. Oral vaccine has a lot of importance, particularly in aquaculture, because you're dealing with millions of animals of different age. You can't go on injecting for a low-priced fish. Okay. And conventional oral vaccine, including human vaccine, animal vaccine, they have a drawback. They get destroyed in the stomach, doesn't reach the immune responsive sites, and poor protection, yeah, unstable protection. That's why oral vaccine is best, but they get destroyed. So what we came up with is, next one, please. I came up with the first time in the world, we came up with a biofilm oral vaccine, making bacteria to develop a biofilm. Biofilm, as you know, it's very resistant to antibiotics or any chemicals. And in fact, it's a problem in medical implants placed in the body, biofilm development, infection often. Okay, but you can make to develop a biofilm on substrate so that you can get high density. Biofilm is with a glycocalyx coat, so it cannot easily destroyed in the stomach. Okay, and another thing is most of the pathogens are in a biofilm mode inside, and the antigen is a mirror image of the biofilm. Okay, so biofilm vaccine is the best rather than the free cell bacteria which you see in the test tube. When you have in a test tube, it's all free cell, not biofilm. Next one, please. This is the typical biofilm of hydrophila, electron micrography on chitin substrate. After our success in fish, I've invited our veterinary people. They have come up with the 2 oral vaccines, one in poultry, another for in dairy. I forgot with pathogen. I think udder problem on bacteria. Next one, please. And these are the different antigens produced in a biofilm mode compared to the fish cell. Okay, next one. So also this different antigen in a biofilm mode, which is useful as a vaccine antigen. Next one, please. This we tried in fish, very high protection in fish, orally vaccinated with this is 3 species of fish. It works. These are herbivore fish. Next one, please. Antibody titer, the one you saw earlier. This is the protection, how much protection they can give to these 3 varieties. Next one. And we also showed where all our biofilm antigens are going in spleen, kidney, how long it is retaining. That's very important in a biofilm vaccine compared to that. This is immunofluorescence using antibody again. Next one, please. This is the time, how long it lasted in different organs, immunocytes, immunogenic organs. Okay. Next one. Any oral vaccine, another task is it should improve gut immunity, mucosal immunity, body mucous as well as gut and oral wherever it is. Okay, and we looked at the gut immunity in calves. It's a Indo-Norway collaboration work. Next one, we fed the fish with biofilm vaccine and looked at the 2D protein analysis of the gut. Several gut proteins linked with immunity were expressed. Next one. And this is a PCR confirmation of those proteins expressed due to the biofilm vaccine in the gut. Next one, please. So far, I talked about the herbivore fish. In fish, there are also carnivore fish. The enzyme profile, pH profile is much, much different from the herbivore fish. So I should see whether it works in carnivore varieties. So we tried in 3, 4 carnivore varieties. Result is almost same. Next one, please. Next. Next one. These are all the performs. I'm just comparing in the carnivore fish. Same type of result, but for the carnivore fish. Next one, please. This one technology is transferred again to Urbac Animal Health. It is undergoing field evaluation. Next one. At the end, before I retire, why I'm saying retired, there's a lot of scope for you youngsters to take up the work. There are some parasites. It's very, very difficult to design a vaccine for parasite because multicellular parasites, very difficult, different life cycle stages. People are struggling, but nevertheless, there are few successful vaccines in veterinary and also in fish. Somebody has come up with a vaccine parasite. Like we have a Lernia, which if you look at, you don't see in the market, you don't eat fish again. Next one, please. These are the various life stages of the parasite. Look at this. Very, very complex. To develop a vaccine, still now we don't have successful vaccine for malarial parasite or trypanosome or whatever it is. Very difficult because of the antigen, number, complexity. Okay, next one. We resorted to reverse vaccinology technology. Like conventionally, we go using whole organism, bacteria or virus, and then going for subunits, looking at the performance, and it takes 7, 8 years. Luckily, we got coronavirus in 2 years. Otherwise, most of the vaccine are 7, 8, 10 years. She knows. Reverse vaccine technologies, in this case, like you're looking from the top, taking the transcriptome in the parasite infected fish and also the various parasite stages, then comparing that to immunogenic and vaccine antigen, then go for there. You are saving a lot of time. Next one, please. We went for transcriptome of adult larval stages of the parasite, because this is very important. Again, most of the agriculture pesticides are in our farmer ponds. That's why I said, I don't feel like eating fish. People just dump to get rid of the parasite, and it is developing resistance again. Okay, next one. This is the one. Okay, so we did some study, next one, and came up with some putative antigen scope for developing vaccine. And I retired there. I left it to my juniors. I don't know where they are. Okay, next one. These are some of the technology we transfer in diagnostics and oral vaccine. In both, we have transferred few technologies. Some are undergoing field studies. Next one. We commercialized diagnostic Rapidot. I showed you earlier. Next one. Next one, go fast. These are publications in various, most of these in international journals, about 80 or 90, about 60 or 70 international. This one you can skip. Next one, please. Next one. A list of publications, that's all. To the youngsters, because I don't know, we are just improving in publication. We are very poor in publications. Next one. Next one. Next one. Next one. This is the acknowledgement. ICR, DBT, International Foundation for Science, Sweden. Many youngsters here, I got IFS project for 12 years. It should be less than 40 years. I think there must be some agriculture faculty as well. Very good. Many people don't know. Now they have raised the age. I don't know why. China and India, they have raised the age. And ODA, European Union, BBSRC, UK, and UNESCO. These are our funding support. Of course, I should not forget my universities, 2 universities. People always grumble, no. We got very good support. Only things you'll have to fight. Finance people create problem. Otherwise, faculty, no problem. It's all what evasive answers. No, I took up the one research. Youngsters, please bear in mind, there's a lot of scope for research in India. Okay. And the take-home message is, go for farmer level or common man level diagnostic kits in India. Like there are so many pregnancy kits, that, this and all. it has got a greater impact. Okay. Next one is oral vaccine for not only human beings, animals also. You can go for oral vaccine. It is approved. Our veterinary people have come up with 2 vaccines. I think this is all the one I just want to present. Thank you. I think I ran fast. If you are interested to see this cassette, it's very, very simple cassette.
Hello. Thank you very much, sir. It is probably a very excellent talk, rather excellent, very healthy talk, rather. The topic is very pleasant for this morning. In fact, not only the agriculture, for all agricultural activities, the health management is most important. And as the agriculture article, every agriculture farming is getting intensified, so is also aquaculture. And one thing about aquaculture is that from the capture, as the speaker himself, so it is getting into the culture activity. When there is an intensification of the culture, there is a management, lot of management problems with there. One of them is to control the diseases. And when you are looking into the controlling the diseases, probably we are all aware of that, particularly in the ecosystem, they'll be dumping a lot of drugs and so many things in this direction to safeguard the environment and to safeguard the consumer as well as the water bodies. It is probably best way to control the disease through early diagnosis through vaccination and treat them. In that direction, this is a thought-provoking talk. In addition to that, as the speaker himself has suggested, health management, either you're from agriculture or from animal husbandry, the basic principles is there. Probably this talk will give you insight for your future development in this direction. With this, I'll open the topic for the question. Any clarifications, rather?
Yes. Yeah.
Excellent talk, sir. I have one question. Oral vaccines are very difficult and you don't get that level of immunogenicity. And
Level
looking
of?
oral vaccine,
Yeah, yeah.
because there are so much of acid and all, by the time it will release, you don't get that, what the type of protection you wanted. And looking at the fish, that aqua. So have you ever, people are thinking of mucosal vaccine, you just put in the pond and it will absorb to the skin and then it
Oh,
can.
immersion vaccine again, uptake is not much. When you go for, people have tried with virus, antigen, bacterial, in a osmotic solution, but it is a lot of work, stress on the fish. This oral is very, very simple, through the feed. It goes through the feed.
Okay. And then regarding that monoclonal antibody based diagnostic, so do you see type of cross reactivity? Is that
Oh, you check
it's,
with,
it's a very naive question. To the particular type of virus, is that different strains? And if you use that, do you see the cross reactivity with different strains, or it's very specific?
no, when you go for a monoclonal, more often you get a specific monoclonal. Okay, correct. And there are viruses sharing similar epitopes. I'm talking about epitopes
Yes.
within the antigen. In that case, you can sort out with immunodot or whatever test, whether they are matching or what. I have worked with the IPN virus, I said, and there's a lot of serotypes. So I have checked the serotypes using a panel of monoclonal antibodies. 7 monoclonal antibodies, 7 similar serotypes. Test, you get the picture.
Okay, and all these maps, you said that it has to be stored in the 4-degree. So
map kits, yes.
map kits.
Yes.
So there are also paper-based dot bot technology that can be dry.
There's the lateral flow one.
Okay.
It's what? Already, what is the term like? Adsorbed to nitrocellulose.
So
ready
these
to
are
use.
more stable, thermostable?
Yeah.
Is it?
Okay.
Thank you. Thanks.
But any lateral flow, sensitivity is much low. It has to flow laterally, and micro pore size is bigger compared to vertical flow.
Thank you, sir, for this interesting talk. I wish I could have spent some more time on that. Anyway, you talked about the vaccination and development of these antibodies and all. So, of course, you told that so much misuse of antibodies are there, and we afraid of eating that
Antibiotics.
antibiotics. Sorry. So, with this vaccination, also, if any there long-term effects on human consumption, anything like that.
I don't think. I don't think. But the other side is some of the countries which are experts in formal culture in Norway, they have using only vaccines for viruses and some bacteria. They have totally stopping the antibiotic use. But as you said, antibiotics, I mean, vaccine use on human consumer, that's your question, right? That I think it's time only answers. I don't think. Right? Yes, please.
Excellent talk, sir. This rapid dot technology, what you had licensed to Virbac,
Hmm.
is it an exclusive or non-exclusive license?
I think it is exclusive.
Exclusive license. Okay, sir. This antibiotic things also
Antibiotic not yet transferred.
not a transfer.
We went for field trials, corona started.
Okay.
Okay. So we are caught in.
Okay, sir. To whom we need to communicate, sir, if we are interested in any of the things?
We have to see. We have to see. Now it's difficult to deal with. Like I'm in Shumaga, away from my university, my client, whoever it is.
Okay.
But I'm trying.
Okay.
And meanwhile, we had difficulty in storing the clones because the
Okay.
corona,
Okay, sir.
liquid nitrogen, Mangalore liquid nitrogen supply is not like in Bangalore. I stored some in our IHEB. Okay.
Yes,
So revival and next stage.
Okay, sir. Thank you.
Right. Thank you.
Thank you very much, sir, for enlightening us on fisheries. Now I request our chairperson to hand over the memento. And please join co-chair and even rapporteur for this.
In fact, it is proud moment for me. Is also my teacher.
Not my student was
The next speaker is Dr. Sweetie Samal. Please come over to the dais.
Dr. Sweetie Sama has received a PhD in molecular virology from the University of Maryland and currently working as principal scientist at a Translational Health Science and Technology Institute an Autonomous Institute of Department of Biotechnology, Government of India. She has over 15 years of research experience in the area of infectious diseases biology, working on viruses such as paramyxovirus, influenza, HIV, dengue, and SARS-CoV-2. Her research major focus areas are influenza, SARS-CoV-2, and dengue viruses. Her lab is extensively working on SARS-CoV-2 since the onset of the pandemic in 2020, mainly focusing on virology and vaccine development. Their group has developed spike-based pseudovirus assay, live virus assays, and various soluble proteins from the structural spike protein, and it's a domain in a mammalian suspension culture. She has various grants from the Department of Biotechnology for the development of influenza vaccines, the Bill and Melinda Gates Foundations and BARAC. On the virology domain, her lab is to understand the different tropism of mutant viruses and the entry and fusion mechanism. She has developed an interferon receptor deficient AG129 model to support the R&D of arboviruses. Our lab is extensively working on developing novel vaccine candidates against influenza, SARS-CoV-2, and dengue. Thank you.
Thank you, sir. Very good morning to all of you. And before I begin, I would like to thank you all for this excellent conference. And I thank all the organizing committee and mainly Professor Bina for giving me an opportunity to be a part of you. So today talk, I will mainly focus, I will take you through the human viruses of genetic and emerging with pandemic potential and how the vaccine helped us protecting since our childhood. But still, what are the major bottlenecks and how India can come forward to the global platform in making the novel vaccines against these emerging viruses? Okay, so I think if you look at the slide, most of us heard of these viruses: influenza, dengue, HIV, Ebola, Nipah, monkeypox. How many of you have heard monkeypox virus? Just, okay, now everyone. Have you heard it, 2 months back? That is a monkeypox. Whoever has born after 1980, we have not taken smallpox virus. Okay, the smallpox has been eradicated. In 1970, the smallpox vaccine has been stopped. That means the youngsters are now prone to the pox virus, right? And it's a very bad virus. So if you look at the slide, from the beginning of the human race, we are closely associated with the animals, feces, and sir now has said that, the fish has so many disease. So more close you stay with this animals or the plants or the environment, the human has more chance to get new viruses because we are notorious. We don't, follow the nature law. We manipulate them and then we get affected. So the similar thing has happened in SARS-CoV-2. I think we all have heard the SARS-CoV-2 virus has come from the bat, and it's a novel coronavirus. It's not novel. In 2003, we had another outbreak, but that time WhatsApp and all this internet facility was not that. That's why nobody knew, so much that we knew today. But coronavirus family has caused pandemic in 2003. It is SARS-CoV-1. It is called severe acute respiratory disease 1. And then another virus called MERS, Middle East respiratory disease. It is the intermediate host is the camel. It also causes severe disease. So in the coronavirus COVID-19 pandemic, out of 100, only 2 people die. So the disease survival is mortality rate is only 2 to 3%. But for the SARS and MERS, the mortality rate is 10%. That means if it infects the 100 people, 10 people will die. Okay, and we don't have a vaccine for SARS-CoV-1 and MERS. That's why it's not development of vaccine, it's not easy. And people are struggling. So if you look at the slide and you can see that even though the source has been thought that it has come from bat because the sequence has a lot of similarity with the human, but it is also in hypothesis that there may be some intermediate host like pangolin or snake. And it has been shown that this new virus can transmit to cat and ferrets, to duck, to canine ducks, domesticated ducks, and the tiger. We have 2-3 cases in India where we are seeing the coronavirus in zoo. That's why people from the veterinary department, now they want to develop vaccine against the animals. So just think that if these two mix and you get a new virus, then what will happen, right? That's why, and the vaccine has been developed for this COVID-1 virus, not for this virus. So that's why the virus has been constantly emerging. And you have around 208 viruses which have genetic importance. And out of these, if you talk about the India, so some of the major diseases are rabies, Japanese encephalitis, Nipah virus, which is mostly seen in Kerala, Northeast. There are Cassinoid forest disease virus. This is one very bad hemorrhagic fever virus. We have chikungunya, dengue, influenza, and now COVID. So we are not talking about HIV because it is not a genetic important virus. So out of the 4 people over the global, 1 people die because of the genetic disease, and the death rate of zoonosis disease is 2.7 million. So, and the only good intervention, what we have also understood, is the vaccine. So if you look at your vaccine card from the childhood, we all have taken this polio MMR. Just think that if you have a world without vaccine, maybe everybody till now, we would not have reached that 7.6 billion people. Maybe half of the population has wiped out if vaccination was not there. And I heard there is a question that we are taking the vaccine, whether we will have the residual. No, we don't get the residual from the vaccine, but there are different kind of vaccine platform. So I'll talk about that. So what is a vaccine? So if you talk about the vaccine, the most active ingredient is the antigen. So getting an antigen from the virus or bacteria or the fungi is the most difficult one, which can produce the antibodies in your body. Once you get it, along with that, people give adjuvant. The adjuvant is mixed, which can stimulate your T cell response. Okay, so we all have read biology in 12th and in our graduation. So there are 3 arms in a body. First is your innate. When you get a pathogen, you first react. That has inherited from your mother. It's a genetical. Then there are 2 response. One is the humoral response. One is the T cell response, adaptive response. So humoral response is the vaccine, which we all are focusing for the antibody. Then come the T cell response. To get the T cell response, adjuvants are incorporated in the vaccine. Rest, more than 70% you have the water, but to make the vaccine stable, you need stabilizer or preservatives. And there are some residual traces, but for the regulatory concern, usually when the vaccine comes to the human, because there is a very strict rule. And as I sir said that it takes 7 to 10 years or 30 years to get a vaccine. And COVID is the only vaccine which we got in 256 days because it's a big pandemic. It's a pandemic in the millennium. Usually the vaccine takes nearly 10 years to develop because the regulatory, as it is coming to the human, so you have to be very concerned that none of the residual traces should be present there. So these are the common vaccine platforms. So this is how many of you have taken Covaxin? So what platform is that? Can you tell me from here? It's an inactivated vaccine. How many of you have taken the Covishield? Okay, so can you tell that what is the platform? No, it's a viral vector vaccine. Okay, and then recently we have got approval for the Corbevax. It's a subunit protein-based vaccine. So these are the major platforms which is available, and we didn't get the Pfizer and Moderna, which are the best vaccine till now. This is the mRNA-based vaccine. Okay, so and the adjuvant which is mostly used, whether it's a animal or the human vaccine, are the aluminum, emulsion, some of the TLR agonist, and very recently people are working on the sting agonist. So I will not go into the detail, but for most of the more than 90% of the vaccine, we always incorporate some adjuvant so that you can get your memory B cells stimulated. So today I will mainly focus on the coronavirus, which is a very large family of the virus. It's a RNA virus. So making a vaccine against the RNA virus is also challenging, and I will tell you why. The next is the influenza virus. So our lab mainly working in these 2 viruses, and we are mainly interested for rabies virus now. We all know that we have the vaccine for the rabies, but there are a lot of complications. So now the scientific community is moving forward to improve the vaccine from the current vaccine. So we are very much interested for this development of this rabies vaccine, and we'll be happy to collaborate if people from animal sector would like to come forward. So this is the COVID SARS-CoV-2 structure. So it's a very spherical large structure, and this is the RNA genome, and this crown-like molecules is known as the spike proteins. And because of it decorate the whole virus surface like a crown, it is envelope virus. And because this spike protein comes out of the envelope, this was first recognized by the host. That's why all the virus, the vaccine which you are taking is based on this spike protein. So as I told you, so we also have some DNA vaccine, vector-based vaccine. This Sputnik V, Covishield, Janssen, all these are vector-based vaccine in which you take a vector and where you incorporate the spike protein. You have the killed inactivated vaccine where we have taken the whole virus, not the spike protein, the whole virus. You kill the virus either with formalin or HIT or ether, and then you use the whole virus as a vaccine. You have the mRNA-based vaccine, which is a very new technology, and it is very well worked for the COVID. And then we have this protein subunit vaccine. So there are some side effects, but we won't go into that. So this is the schematic gives you the idea like how the vaccine has been developed and how it is worked. As I told you, they have taken a live virus, which is called adenovirus. So we all are affected by adenovirus. So if I take the human adenovirus, I already have the immunity against the adenovirus. So that's why my vaccine will not work. So what they have taken a chimpanzee adenovirus. So from the chimpanzee adenovirus, they have taken out this adenovirus envelope protein. Now they have introduced the spike SARS-CoV-2, this spike protein to this live virus. And this live virus, they have attenuated, it cannot cause the disease. It's a single time. So when you inject this virus, the cells express the spike protein. So these are the spike proteins. So then the antibodies are produced against the spike protein. And if you get a virus infected with a virus, then your body protects us. So now what's the problem we are facing now? Because the it's a RNA virus. Okay, so I think all of you must in the plant, you must be studying, right? How the cells replicate with the DNA polymerase, RNA polymerase. So the virus, because it's a RNA virus, so it has RNA polymerase, which doesn't have proofreading mechanism. That means it can mutate, it can introduce new mutation. That's why all the vaccine has made from the Wuhan strain in 2020. After that, in 2021, we are stuck with Delta. I will not go through the alpha, delta, beta, gamma. It has come from, it has first seen in London, but in 2021, we are first stuck with Delta because of which a lot of people died. Okay, so you already have a virus for the Wuhan, you already have the antibody, but it didn't protect the Delta. And now we are having Omicron. So the vaccine, that's why even though you have taken the vaccines, still you are getting the disease, but the disease severity was less because some level of cross protection you are seeing. But still, if you look at the newspaper, you will find that every day, there are maybe 14,000 people are infected with COVID, and out of that, 50 or 100 are dying. Why? Because they are getting infected with Omicron, and the body, in whatever the reason, maybe the vaccine is not protective, maybe the body has run out of the antibodies. That's why it is not able to protect. So that's why you need a vaccine which can, even though new emerging strains will come up, but still it can protect you. So that's why now the next generation of COVID vaccine will be a vaccine which can protect from the universal coronavirus. That means if you take the vaccine, it can protect you. Omicron, suppose after 6 months you get a new virus, it can still protect you. So for this, what we have done, we have developed one plug and play platform in which we have taken a non-structural protein of the SARS-CoV-2. It has a property that it can self-assemble to form a nano cage. That means a bigger cage. So here you can stitch different spike or domain of different viruses. And when you then you develop it, it is a bacterial expression system, bacterial subunit based protein. So it's still in the developing stage, but it's working fine as a proof of concept where we have stitched Delta, Alpha, and Omicron, and we can get the protection against all the three. And now we are trying to develop for the beta coronaviruses, which consist of COVID-1, MERS, and COVID-2. Okay, now coming to influenza. So every year, every season, we get infection with the flu. But because the Indians have very good innate immunity, that's why the death rate is less here, or we don't count. 10,000 people die of influenza every year, but our epidemiological survey, it's not that good. And in the national immunization, we don't have the influenza. But if you look at the Europe and US, every year there is more than 250,000 people die because of the influenza infection. And the virus is present in everywhere. I mean, it's in the fish, in the seal, in the domestic animals, in the avian, in the poultry, in the swine. And I think most of you must be heard of avian flu and swine flu. And now we are also seeing it is from the bat. And for the COVID mainly infected the old people and the vulnerable people. The children were skipped, but for the problem with influenza that it can infect the children and it causes heavy mortality in the children. So actually in 2018, we not celebrated, we remember the 100 years of the Spanish flu. Okay, and that's why the world was preparing and they were expecting that the next pandemic will come, the influenza, because every 10 years we are expected now to face a pandemic. The last pandemic we faced in 2009 with H1N1 swine flu virus. But unfortunately, it came the COVID. So now we are preparing whether if the influenza virus will start, and it's a very bad virus, unlike COVID, where death rate can be very high, then what will happen? So I will not go through the details, but I will only tell, I will only, ask you to just see how the genome is organized. The COVID, you find a single genome, but here is the 8 genome. And because of the 8 genome, it goes through antigenic resortment and resortment. That means if you suppose this is the swine influenza, this is avian influenza, both will come together and these segments will interchange. Then you get a new virus. So it is called antigenic shift. And because of that, from 1889 to 2005, every 5 and 10 years, we are facing some kind of new influenza outbreak. So this is an schematic showing that how this shift and drift occurs and which can lead to the pandemic in the human. So for the influenza, vaccines are available, but first of all, we are not aware unless and until somebody, I mean, the disease is very high, we don't go to the doctor. That's our, inherited attitude. So that's why we don't take the vaccine. So even though the vaccines is available, but still the vaccine is not effective for the children and for the old. So in our lab, we are mainly focusing on this two structural proteins, which is the HA and M2, to develop mammalian-based vaccines. So this is HA-based vaccine, and I will not go through the detail, but this is developed in the mammalian expression system. And this very recently we saw that in the mouse challenge model, it is giving complete protection. So it can be used. So now we are in talk with the industry and patent and all kind of things, whether we can take it forward. This is another kind of, vaccine development, which is also a mammalian base, and that can also seeing very good promising as a vaccine candidate. So in our lab, we are also focusing on this two vaccine candidates. So Rabies, I won't go through because all of us know about the rabies. It's a nervous disease. It's a disease which can affect our nervous system. This is a kind of typical vaccine where you take the vaccine after the exposure, not before. For the COVID or for other viruses, we are taking the vaccines in our childhood or in our adults so that if the virus will infect, we can protect us. But this vaccine we take when we get infected with a rabbit or cat or fox infected with rabbit. So this is called pre-exposure, post-exposure vaccine. And this is showing, that how endemic the India is for the rabies. Again, I won't go through the detail, but the vaccine we are using are the killed viral vaccine, and most of the time, it's not very effective and the efficacy is going down. The price is very high. So that's why now we are trying, the scientific community is trying to develop a subunit based vaccine. So here also it's a RNA virus, and you will see every time we are mainly focused on the structural protein. So even though the virus are different, there are some kind of conserve remains in the structure which help us to design the vaccine. So this is the last slide. This is also a trimeric protein similar to kind of SARS-CoV-2. And but here you cannot make the vaccine in another expression system because it needs the native-like structure. So that's why the mammalian expression system is the best. So we'll be happy to make collaboration if anybody is interested for this. With this, I would like to acknowledge this is our lab. We got several funding from CEPI, European Union, and Department of Biotechnology and BIRAC. And with this, I would like to thank Department of Plant Biotechnology for giving me an opportunity to discuss and to share with you about viruses and vaccines. And I'll be happy to take any question. Thank you.
Thank you, Sweet Shamar. It's a very good. And we are very well exposed to the virus. There are a good number of viruses, and we need to be protected ourselves. Probably in this direction, she has highlighted how exactly the process of vaccine production is very important. Particularly, that is more important to the younger generation sitting in the backside who can think of working in the vaccine with the vaccines, and the very good information for the youngsters. In addition, that apart, you have mentioned the presentation about the one good number of times about the COVID. All are exposed to the COVID, whether it is really from the Wuhan or from the cave of the Wuhan. And how exactly it has been confirmed. There are some contradictions. That COVID virus, there are some discussions, arguments, counter arguments. It is from the Wuhan's biosafety level 2 lab, somebody is telling, and some of them, no, it is from the wet market. Somebody is telling it near to the wet market, there was a cave of bats from there. So what is you are discussing in the scientific area?
I think as a human being, we always like to look at that, the cause or the solution rather than looking at the cause and solution. You always go to. I completely understand. Look, it can be sometimes China. Okay, it's this platform. We should not talk about that. But these viruses has to be that viruses can be divided into 4 labels: biosafety 1, biosafety 2, biosafety 3, biosafety 4. Your polio virus comes as a biosafety 4. So think about that. And it is a bio weapon. If somebody can intentionally, if it is brings into the environment, it will cause a cause, and you need lots of money, for the vaccination drive. It's not easy. Crores and crores of money you need to do the vaccination, right? And because it was a pandemic, all the country came forward. So if you look at how you know that it has come from a lab, or it has come from a wet market. Okay, so how you know? Suppose maybe you guys are in the plant, there are 2 type of strain. Okay, and suddenly you find a new strain. How will you do it? You'll go for sequencing, right? Okay, if there is any mixture of anything. So the sequencing result says it has the bat sequence, and the Wuhan wet market also has all kind of animals. Wuhan lab is one of the best lab BSL-4 which has been developed by the US, but they also do the bat research. Okay, and China is a very cold country. Okay, so and it if the lab accidents can happen. Okay, but so there are a lot of controversies going on, whether it has come from the lab, from the wheat market, whatever the cause, it has come from the bat. Okay, and usually bat, they just stay in the up and they stay in the dark. All our temples are full of bat, right? But you never get any infection in India. But why suddenly you are getting this Nipah and all? Because we are encroaching. Think about 20 years, like, sir, in your childhood, how many people are going to a temple? Maybe 100. Today you go any day, there will be 10,000 people, okay, in the temple. But the temple remain that small. The encroachment, the bat, their life, their space remain the same, but people are coming in. That's why you are coming more closely with the bat, fecal matter, with the bats. So for your question, this is a very tricky, because nobody knows. We can make the assumption, but it has come from bat. That is for sure. And it has never come before. Only virus which has come from bat is the Nipah. Okay, now we are getting in India.
Madam, I have a couple of questions. We are fortunate to come from Professor Ramanjanagoda lab. It was known for vaccines. Particularly, we worked on vaccine and we expressed in yeast as well as a subunit vaccine, a glycoprotein gene in collaboration with Nimans. And also off late, bioinformatics study has been conducted on SARS through our lab. The receptor binding domain has been considered to produce a universal vaccine.
No, it's
No. As per the reports, we have studied RBD region of 3192, 541 amino acid sequence. When we compare this sequence with all the variants,
yes,
there are 7 places at which mutations are there. So how best we can use this RBD to produce a subunit vaccine for universal?
we can't, because the RBD in the Wuhan, there was no mutation.
Yes.
The first mutation came A501.
Yeah.
Okay, and the RBD binds to the human AC2 receptor.
yes, too.
Okay,
Yeah.
so if you look, you can compare how much the binding happens in the in vitro label. So if you take a soluble RBD of Wuhan, bind to the HSC2, suppose you are getting a 0.1 value, suppose you are getting a value 1, you can do the method is called octet or SPR, and you take a RBD of alpha. RBD, the alpha contains only 501, no other, only single mutation. It changed the structure completely. Now you bind with AC2, you will get the binding affinity to 9. That means 9 times affinity it bind to the AC2. So when the virus goes, suppose, just imagine, it takes for Wuhan to enter into the cells, it takes 10 minutes. Within the 10 minutes, your body will do all kind of defense. But for the alpha, if it is take 1 minute, by the time your body comes and defend, already 10 viruses are already inside. So how your body will defend? That's why the RBD can't be used. The single RBD can't be used. Your question, the RBD is the receptor binding domain, but for every virus, the receptor binding domain changes. So for the subunit based vaccine, you will go with one RBD sequence, right? Either you will go with 501, or you will go with Wuhan, or you will go with Omicron, which is now 7. But suppose tomorrow there is another virus come, Delta Cron, which will have 3 mutation, and again it changed the structure. Then your Omicron RBD will not work against that.
What would be the solution now to produce a?
That's why you don't get a vaccine for HIV. For the 30 years we are working, we don't get a vaccine for dengue.
And you say it is variant specific vaccines we have to
It's
rely
not
on?
variants. Company works because they want the money, right? And now we also know that after the virus infection, also if you don't take a vaccine, you have antibody. So pushing people to get the booster vaccine. How many of you have taken booster vaccine? Just see, I think only 10%. Because other people think that, we think that we don't need a booster. So Every year you cannot make vaccine and spend crores of money where there is no takers.
Then what will be the final
That's
solution?
what the solution is, that you have to bring out some noble platform, think out of the box so that you can stitch multiple epitope in one candidate. In one go, you take the conserved epitope. In the virus is very smart. It's just one gene. This virus is 11 kb. The length is 11 kilobase, the genome, and the human is 10 to the power 9 billion kilobase. Just think that your virus can come and kill us. That means think that this tiny organism is so smart. So you have to take the conserved reason and put that conserved reason, do some stapling kind of thing so that you get a universal vaccine which can protect you irrespective of any virus come. So that should be the another approach.
Stitching of all RBDs of all the variants
All
in
the
one go?
or not all the variants you cannot express. If you stitch RBD is around 300 amino acid. If you stitch 5 RBD, it will be 15 amino acid. If as a company you need more production so that antigen value will be less. So if you go higher molecular weight, your antigen label will come down. So your cost making will not, so everything you have to calculate. That's why vaccine is difficult. It's very difficult, at least because the way we are getting smart, the virus is also getting smart.
What could be the constructive solution for this? Because
Constructive
you have done a lot of
solution
research in your.
We have to, like people, we have to come like this type of platform. Everybody should come forward. We should work together. We should exchange idea, and then we should be, happy to exchange the ideas, platforms. Like I heard that sir said that, South, I have 10 electron microscope. Okay. And we are struggling to get one structure analysis of spike in North. Right. So we don't know our stake. What are the stake?
Can I
Thank you so much
interrupt,
for your
please?
wonderful presentation.
Can I interrupt?
In your slide on source of infection, human being body was covered all around with animals only. Can you name one virus that comes from plant source?
That you have to tell me. We'll be happy to make any vaccine.
Okay. The follow-up question. Yeah,
yeah.
I know.
It can come after 10 years.
No, but so far there is no single virus that has been known to come from a plant source.
Yeah, because plant and animals, we still belong to the same family, right? We have come from the monkey and orangutan, right?
The follow-up question is,
Yeah.
Are non-vegetarians more susceptible to infections?
No,
Overall,
the thing is,
please listen. Compared to obligate vegetarian people. Even corona, an example where it came from a non-vegetarian source and then it came to.
You know what is the lifespan of the highest lifespan of the mane in the world, whose the country is? It's the Middle East. Middle East mane has the highest lifespan. In India, you will find the male lifespan is lesser than women. Because, but the thing is that, we have progressed. Lot of our thought has not progressed. Like, if you look at the 30 years of our, grandparents, their lifestyle, their thought process, they are not taking that stress. So even they are taking vegetarian, it's all pure. They used to work hard. They used to less stress. Their thought process is very good. They are very simple. But suppose I stick to the vegetarian, but every day I'm taking so much of stress element inside me. Right. So how come I can overcome? So it's a very big debate, but we did one bone density test in our institute, and we found that vegetarian people has very less bone density when they reach 40. So, so be careful, like youngster, our brain is shrinking, everything is shrinking. So think that, by the time you will reach 60, so what will happen to your bone?
Any questions?
Hi, can I have an answer to the question, one of the question, please?
Thank you, Dr. Sweety. It's a nice lecture after my last lecture in animal virology in 1989. Okay. Like I have few questions like for my own information, maybe it helps others. the ingredients you showed in making vaccine, you said some of them are inert and in no way they are harmful or whatever it is.
Uh, Again, look, because it's a YouTube stream, streaming, it's all my thought. Okay, don't take it anything like for mRNA DNA vaccine.
Like,
I'm a virologist, so if you ask me what vaccine you would like to take, I'll go for a subunit or inactivated vaccine. Okay.
the masala goes with the
Yeah,
vaccine.
I'm not very comfortable with mRNA or DNA vaccine because I
Okay.
don't know or a viral vector vaccine. Even though they are very safe, but you don't know after 20 years. I mean, I'm not very comfortable. That's
No,
lots of
why
genetic.
I'm asking is sometime 1 in a million may have reaction, right? Violent reaction
Safety
because
is
of the
there,
vaccine.
safety
Is
is
it due to the antigen per se or the ingredients?
No, it's not about antigen. It's also about the host. Like out of 100 people, one people may come with some kind of allergic reaction,
Mhm,
right?
mhm.
But then it because of the host also, how the host is reacting.
Immune system.
Immune system is reacting. But because of one person, that 99 people is being saved.
Oh.
That's why you can't just, just think about polio. Polio vaccine, if you're not given to the children, there was cases where, it has caused some infirmity, leg infirmity on some children. But there are millions of children have, survived measles, mumps, chicken pox. So all that this children vaccination. So it saved a life. But there are side effects. Because this COVID vaccine within 300 days, we didn't have any option. We wanted to save the people. So, but there may be slowly, we are again, not because of the vaccine, because of the virus also. Maybe the heart, it has changed. People are getting more heart attacks these days after the COVID.
This information for all. Like my daughter is 31 years old. She missed some of her childhood vaccine. Recently, she had vaccination in Bangalore. One vaccine I forgot. She's in US now at the moment. Violent reaction within 1 minute. She couldn't breathe and her face swollen up. All the doctors came running. This happened some 6 months back.
Yeah.
And finally, they could give some treatment. I don't know what it is, but we could save her.
Yeah. So that's the problem.
1
with-
in million, billion.
Yeah,
no,
because one of the problem with the virus, they also cause antibody dependent cytotoxicity. That's the problem because which we are not able to do the dengue vaccine. So dengue has 4 serotype, 1, 2, 3, 4. Suppose you have one, then you infect with the 2. So you will say you have one dengue, so your antibody is there in for the dengue. So now you are get the 2. So you think that you will be protected. Dengue 1 is asymptomatic. Okay. But when you get the 2, instead of, killing your viruses, this antibody bind to the T cell cytotoxic receptor and enhance the cytokines. That means you get more fever, more dengue hemorrhagic fever, and you die. That's why, we are not able to produce the dengue vaccine, even though we are trying, and there the reactivity is much more higher. This COVID vaccine, that Covishield and Covaxin, was quite safe because the spike protein is not that, mutating, it doesn't causing ADCC. But SARS and MERS is causing ADCC, even though they are the same brother and sister from the same family. So COVID is good. So we are little lucky.
Okay, in your platform, can we have a gene edited vaccines like? I mean, is it?
No, that CRISPR technology people are trying, but
Okay.
still, because it's not about the technology. You have to take forward for the company, and when you put in a large production, lot of things fail.
And somewhere you said the nucleic acid vaccines are better for corona.
mRNA-based vaccine, it worked for corona.
What is the reason like?
Because for the flu vaccine, as I said, it has lot of strains are there. So you cannot make vaccine for one. Spike is, even though there is variation, but still it is conserved. That's why the initial mRNA vaccine, it's a whole spike protein vaccine. Okay, so when you give the mRNA Moderna, the antibody titer came very high and it is lasting longer than Covishield or Covaxin.
Okay.
Covaxin, whoever has take after 4 months, you may get the virus infection because it is not lasting. Like suppose you take tetanus vaccine, it's not a viral vaccine. So the doctor will say that after 10 years, you take another tetanus. It stay for 10 years. But this doesn't. So, virus and vaccine is very interesting, very complicated. It may take whole day. I don't want to.
if there is time, a very two short question, information for all. Like through genetic engineering, have we created deadly virus anywhere in the world?
It is not
So,
accepted.
because there's
We-
so much of a discussion about the corona, Chinese genetically engineered, and that, this and that.
Yeah, yeah.
I don't think it is.
No, people like sometimes accidentally you do. Like suppose, like any vaccine I need to first do in the animal study, then take it to the human.
Yeah.
So for to do the preclinical animal study, you need the challenge virus. Sometimes the human virus, they don't replicate in the animal. So I need to passage, adapt it to the animal. So during the adaptation, some mutation happened and it became more virulent. So one incident happened in influenza in Netherlands, for which the lab then later on was closed.
Okay.
So
The
this
last
happened.
question anyway. Rabies shot we take soon after the bite.
Yes.
How fast it is in antibody production? Like instead, can we not take it well in advance like other vaccines? I mean,
That's the thing. If you can make a subunit based good vaccine, you can take it.
in advance, like
Yeah, you can take it in
anticipating.
advance. That was like people who are working with the dogs
Veterinarians.
and veterinarians, they can take. That's what the next approach. Like if you there are giving the rabies shot is also two types. If you have already anytime have taken rabies, then they give you two shot. But if you are naive, no rabies vaccine at all in your lifetime, then three shot, 1, 0, third, then 14 days. Then only your immunogenicity boost up, and then even though your virus will not able to reach the nerve. Once it reach the nerve, you cannot, so it takes 2 months of time. The virus incubation time. I mean, in the COVID, it is 5 days. That's why within third day you get lot of fever, sweating, backache. For the rabies, it after 2 months you will see that your saliva is coming. So you have 2 months. Yeah, that is the more the if you get the bite, you just see now why here is the virus receptor. Virus has the tropism, right? So the tropism is in the brain. For COVID, the tropism is in the heart, I mean respiratory system, right? So if you get a bite in the leg, it will take some time, but if you get a bite here, it will take reverse this time. So based on that. Thank you. Thank you.
During the tea time, we'll continue with this discussion. Now, we'll give an opportunity to the next speaker.
Thank you.
Thank you, ma'am, for very nice presentation and discussion. Now I request our chairperson to hand over the momentum. Please, co-chair, join the co-chairman and rapporteur. Thank you, sir. As the time is a shortage now, there will not be any break for the tea. Tea will be served inside the auditorium itself. Now, I request the next speaker to come over to the dais, Dr. Ravi Manjitaya, please. And I request co-chair persons to introduce the speaker.
Dr. Ravi Manjitaya received his PhD degree studying transcriptional gene regulation from Indian Institute of Science. He did his postdoctoral training in the autophagy related pathways at the University of California, San Diego. Before joining JNCSR in 2011, he was a Wellcome Trust DBT Intermediate Fellow between 2011 to 2016. His lab is interested in understanding the mechanisms that govern the autophagy and related pathways in health and disease. He has received the CDRI award in 2020 for excellence in drug research in life sciences category and S. Ramachandran National Bioscience Award for career development by the Department of Biotechnology, Government of India. Thank you.
Thank you very much. And before I start, I mean, it's a bit of a homecoming for me. 97 December, I came as a master student to GKVK campus here and visited the microbiology department. And that was my first foray looking at the beautiful campus. And while doing my PhD in the common coffee places, I was fortunate enough to keep bumping into Veena. I don't think we ever spoke, but she's been very nice. And I really thank her for giving this opportunity. And we look forward to working together in the future. So I'm probably one of the closest scientists you'll find. Right across the street is Jawaharlal Nehru Center for Advanced Scientific Research, where I'm an associate professor there and I have my lab. And we study a process called autophagy, and I'll talk to you a little bit about this process. It got the Nobel Prize in 2016, and it's one of the most conserved processes that you will see in eukaryotic systems. Whether you're studying yeast, as shown here, or mammalian cells and a bit of plants. So I don't work in plants, but through collaborations, we publish something about plants, which I'll share with you in the subsequent slides, just to keep interest of the people. So what is this field of autophagy and why it's so exciting for us? It turns out that from all organisms, eukaryotic organisms, let's say except bacteria, and but all evolved organisms such as from yeast to humans, every single cell has a garbage removing property. If a cell is going to live longer, very much like a house, you're going to keep your house clean by removing garbage and throwing it out. I mean, in a good community, you want to keep the entire community and area also clean. So autophagy is this process where every cell, whether if this is the nucleus, endoplasmic reticulum, and other organelles that you know, apart from that, the cell has this magical ability, if you wish, to make such Pac-Man-like structures. So if you're computer gaming enthusiast from the old world, you must have played Pac-Man at some point of time. So Pac-Man goes and starts eating stuff. So very much like that, our cells can create such Pac-Mans at will. And these Pac-Man-like double membrane vesicles can go and capture anything and everything inside a cell. They can go and look, take up some mitochondria, some part of the cytoplasm, some other organelles, aggregated proteins, and then bag them together in a double membrane vesicle called autophagosomes. This bagging is very much like throwing trash in our trash bin, dustbin, but the job doesn't end there. And so autophagosome formation essentially sequesters stuff away from the cytoplasm into these vesicles, but it cannot degrade them or destroy them. It acquires that property by fusing with another vesicle called the lysosomes. As you must know, lysosomes are called the suicide bags. They have these hydrolytic enzymes which can chop up everything and anything inside which comes in contact with them. And this double membrane vesicle, when it fuses with the lysosomes, forms something called an autolysosomes. And herein, everything that is there in this gets completely destroyed. Large proteins are broken down into amino acids, complex sugars into simple sugars, lipids, carbohydrates are broken down to sugars, and so many things can be completely destroyed here. And these individual building blocks are then pumped back into cytoplasm. So not only autophagy is a garbage removal system in our cells, but it's a truly a recycling system. And it is very important for survival during starvation. When there is no food outside for the cells, cells rely on autophagy heavily to get energy and nutrition by degrading unwanted or excess stuff inside the cells. Okay, so this can be also visualized in a small movie if it plays like this. So this is inside our cells, and you can see now this double membrane vesicles, by fusing other small vesicles, will start growing into a double membrane vesicle that can capture mitochondria, aggregated proteins, ribosomes, other organelles such as peroxisomes. And this capturing process results in formation of an autophagosome. Autophagosomes can sequester all these away from the cytoplasmic contents. And this autophagosome can now and fuse, in this case, the yeast vacuole, or in mammalian cases, lysosomes, and the contents will be totally degraded. And these degraded stuff can be pumped back into our cytoplasm to give energy to the cell for carrying out its functions. Okay, so this is the process in the cartoon. And one of the things that is now most interesting in the field, the state-of-the-art field, is to understand how we can control this process. For example, there's an AC here or your fan in your house. You have regulators, right? You want to increase the speed, you want to decrease the speed, you can do that. Similarly, the questions people are asking is that, can we control autophagy, more autophagy or less autophagy? Less autophagy in some kind of cancers where you want to kill the cells, and more autophagy when you want to remove more garbage from your cells. And by controlling autophagy, you can control a lot of diseases and provide a lot of therapeutic solutions. And so I'm not able to touch everything today. My lab works on a lot of aspects of autophagy regulation. We use diverse model systems depending on the questions that we are asking. We work a lot with mammalian cells and yeast cells, but we also collaborate with other labs to understand autophagy, how it is controlled, because it's a conserved process, right? So you can test your hypothesis in many different model systems, including the mice models, Drosophila or C. elegans. And as I told you, we also were fortunate enough to work briefly with a plant model organism. Okay, it's not a model organism, but it's a plant that we can talk about. We look at genetic ways of addressing regulators of autophagy. We also have done small molecule screen. We'll talk about some of the Parkinson's and disease associated with autophagy. We will look at that as well. And latest, we are looking at what makes all of us differently susceptible to diseases, because our genetic composition may be very, very slightly different. And that may affect autophagy speed. So the speed at which my body does autophagy or your body does autophagy, if that is different, it can also result one of us being more sick than the other. So we are trying to understand all these processes. We can look at autophagy as capturing something inside a cell by the model that we have shown here. Okay, and we can use yeast very quickly to screen large set of data to understand, to identify new genes or new molecules. There's also a chemical biology approach. What one can do is to develop an assay where you can look at autophagy going faster or slower, depending upon the small molecules that are present in the media. So you can have a library of small chemical molecules, drug-like molecules. You can add it to these cells and ask them to change the speed of autophagy. And the speed can be measured by a real-time fluorescent high throughput assay. And that assay can then lead to identification of a lot of small molecules that can either slow down autophagy or that can increase autophagy. These are some of the known ones. We went on to screen almost 2 lakh small molecule, 200,000 compounds, and identified a whole bunch of compounds that can either induce autophagy or inhibit autophagy. If for those who are interested how we process, I will talk about that things later. But what do we do with these compounds? Once we know that we can identify such compound that can increase autophagy or decrease autophagy, how do we test them in different ways? So here is a small list of compounds that we have published. These are novel potent inducers of autophagy flux, or these can actually slow down autophagy flux. Okay. In fact, we can then use those compounds to understand autophagy in the first place, because that was our initial purpose, to use these small molecules as tools. You can identify molecules such as these that can, through transcriptional mechanism, induce autophagy flux, or you can have molecules that can inhibit at this particular stage. You can have other molecules that can induce fusion of lysosomes with autophagosomes to generate more degradative power, or you can have another molecules that can actually block autophagy flux by preventing fusion or preventing degradation inside the autolysosomes. So at multiple stages, we can use these small molecules that we identified and characterized, suggesting that autophagy regulation can happen at multiple stages, either from the induction at the transcription level or the final level of degradation of the contents of the autolysosome. Okay. And one of the spin-off of this is that when we identify such molecules, companies that are selling molecules suddenly start selling it like an autophagy modulator. So you'll find several companies that now start talking about compounds that we identified as a reversible inducer of autophagy. So they will start saying this compound is a reversible inducer of autophagy and can be used. For some people, I believe plants is of interest of today. And so just wanted to highlight that in plants also autophagy plays a very important role. And what I'm going to talk about here is applies to almost all model organisms, whether it is yeast all the way perhaps to humans as well. So if autophagy is increased in these plants, if we either genetically increase autophagy in plants, these plants will now have increased growth, seed set, seed oil content, and even nitrogen remobilization efficiency and grain filling. And finally, the plant productivity can also go up. So in multiple plant organisms, this has been shown. But if you have now no autophagy in these plants, they can be hypersensitive. They have slow leaf emergence and premature senescence, and lots of problems are there. So you can imagine that a housekeeping function such as garbage or trash removal is so vital for our homes. Similarly, if such a system is either played around in a manner in these plants, you'll also see a similar outcomes, meaning by circulation is very important. We were fortunate to work with a Sri Lankan Canadian through a collaborative grant to address how we can study autophagy in a plant whose genetics is not known. So it's not a model organism, and you don't know, you cannot do much experiments to study autophagy in it. In such a genetically intractable organism, can we use our small molecules to ask questions such as whether autophagy even happens in this beautiful plant? This is a more of a decorative plant used in, in houses and aquariums and things like that. And it forms this nice mesh around it. And so it's very easy for microscopy to visualize this process because autophagy is such a microscopic process. And by using standard inhibitors, we could show that autophagy vesicles can nicely be visualized even in this plant organism. Okay, this is a pretty challenging work, but apart from the known mechanisms that are published that can control autophagy, our molecules were also able to show similar reaction to understand how autophagy functions in these cells. We also looked at autophagy using a marker for autophagosomes called ATG8, using an antibody in a related plant organism called Chlamydomonas. And from that, we were able to stain autophagy and look at how autophagy can be studied in this plant organism. So if you have small molecule as tools in a completely novel system where normal genetic tools like CRISPR or any kind of transformation and genetic manipulation is not possible, you can use small molecules as tools to characterize a pathway and understand the rate of autophagy that is happening in the cells. You can also select then for plants that can do more autophagy, less autophagy, and see what the outcome is there. Okay, so apart from that, in our lab, as I told you, we work with yeast as well. We also work with two very interesting systems where we are trying to address if we can increase autophagy in a disease state, how it can be beneficial. And there are two paradigms I will talk to you about. One, in when our bodies, in our cells, we have get infections. Like we had the previous speaker talk about viral infections, we can also get bacterial infections. Okay? And in bacterial infections, how autophagy can fight out those pathogens and clear them by degrading them in lysosomes. The second problem is in neurodegeneration or protein aggregate related problems, where such as Parkinson's, Alzheimer's, Huntington's, and so on, you have massive protein aggregation inside our cells. These protein clumps are toxic and they just start killing the cells eventually. So if they can be captured by autophagy and cleared, then that's another way of looking at a disease therapy. So I was telling you about this process. I will quickly tell you what insights we got by doing some of the approaches that we do before we go on to the disease models. Okay. So one of the things that is very important is this molecule, this cargo, whatever cargo is captured by autophagosomes, it has to go and fuse with the lysosomes. And to fuse with the lysosomes, you need large protein complexes that will take these two vesicles, bring them together and fuse them inside a cell. And we have identified that a complex called exocyst and some of its components are important for autophagosome lysosome fusion. Okay. And this we can do using gel filtration chromatography. For old timers who do biochemistry experiments, you will know that you can do gel filtration and identify subunits that are essential for autophagy as well as non-autophagy conditions. But some of the subunits are only required for non-autophagy conditions and not for autophagy conditions. And by doing this selective study, we are able to identify proteins that are important to bring together. So this is a cartoon just to show you that a fishing person will throw his fishing rod and to a long distance and able to grab a fish that is several meters away from the person who's doing actually the fishing. And in a similar scenario, these huge tethering complexes can go and grab vesicles that are there inside our cells at a very great distance, but they are able to bring them together to bring about fusion. So understanding such basic science questions is important in generating fusion. This is not a novel concept. It turns out that everything inside our cell, including vesicles and organelles, move around. And for them to interact via vesicles, you need these large tethering complexes. Here are a few examples of several tethering complexes that participate in cellular trafficking, as they are called, or vesicular trafficking, as they're called. And the one that we are working is the exocyst. And we also think that a subsection of the exocyst complex can come together and form a special complex that is required for autophagy functions. So these are shared components. When the whole complex is together, they do some other function. When some parts of the complex come together, they do a separate function for autophagy. Okay, and I was telling you now about, in the last few minutes, I'll talk to you about what applications one can think about using while they study autophagy. Here is an example where you can see, I hope you can see, this is the green colored is a human cell, and it is infected now with the salmonella or typhoid causing bacterium in red. And you can see in this time-lapse movie, over a period of time, the bacteria just start multiplying quite a bit inside these cells. And the whole cell sort of gets filled with the bacteria. When this movie is playing again, you can see some bacteria gets coated with green color. This green color is your autophagy machinery trying to capture bacteria. But in the system that we have created, we can artificially allow the bacteria to overcome whatever the cells are able to do autophagy. And what I'm not showing here, some of the molecules that we identified allow our cells to do more autophagy so that they can capture more of these cells and clear and prevent infection. So this is a new way of thinking about it. autophagy generally doesn't bother much about, let's say, pathogen like an antibiotic would do. So you have gram-positive bacteria, gram-negative bacteria, and your treatment modalities. But people are now thinking about host-directed therapies. That means you don't worry about the pathogen, but you boost up your innate immunity inside the cells, including autophagy, to capture pathogens and clear them. And in the future, one can envisage either combination therapies or therapies that are mostly host-directed, because that way changes in the pathogens and the types of pathogens you're really not worried about, so long as you have efficient innate immunity system and coupled with autophagy to capture and degrade the pathogens you're talking about. Similar approach is used in cancer, which I will not talk about today, but I'll just tell you that in cancer, what we need to do is to stop cancer cells from doing autophagy, because in many cancers, cancer cells rely on autophagy for getting more food and nutrients. When you cut their supply or you block autophagy in those cancer cells, then the regression of this cancer tumor cells is almost, if not faster, as compared to standard chemotherapy or radiotherapy things. So these are some of the future ways where people can look up. Another aspect I told you about was neurodegeneration, where protein aggregates can be captured and cleared by autophagy. And in many cases, that is not an efficient process. As we age, our ability to do autophagy in our brain cells also decreases quite a bit. In addition to that, the protein aggregates actually block autophagy in many, many different ways. And therefore, when you have an autophagy deficient system, there's very much more likelihood of getting neurodegeneration. Okay. And here is what we have done is to take a mouse model, a preclinical mouse model that can do a bit of autophagy, show Parkinson's-like symptoms, because when you give this toxic compound like MPTP, in the substantia nigra pars compacta region in the brain, you can see it has lost most of the dopaminergic neurons. You can count them. There are around 10,000 of them, but you can use this toxin to ablate some of those cells. And these mice now will behave as if they have Parkinson's. But if you give an autophagy inducing compound like the ones that we have spoken about, along with the toxin, okay, what you will see is that those cells will be able to do autophagy, and as a result, they will be able to clear the protein aggregates and not die because of this problem that we are seeing. But however, we have done those experiments where we wait long enough for the neurons to die, that's in the case of severe Parkinsonism, for example, and then you try to induce autophagy, you will not see that effect. Okay, meaning by, because an autophagy is an intracellular process, and if the cells have already died, it's unlikely that there's going to be any autophagy that's going to save you. So it's an early stage way of thinking about host-directed mechanism to clear protein aggregates for looking at saving. It also turns out that from our perspective, autophagy that happens in yeast cells versus autophagy that happens in our other cells versus the autophagy that happens in brain is very, very different. You know, in the brain, for example, the neurons are extremely large cells, and they can have, they can be 1 meter, 2 meter, 3 meter long neurons you could have. So how does autophagy go about capturing, clearing, degrading everything? Turns out very interestingly that unlike most of the autophagy that people study, which happens near the nucleus or in the cell body or the soma, as they say it, autophagy generation actually happens more at the synapse, it turns out. So presynapse is a place where more autophagy happens. And it also turns out in neurodegeneration diseases such as Parkinson, Alzheimer's, the degradation or the degeneration starts at the presynapse. So one of the questions that we are trying to address is that can we locally induce autophagy at the presynapse so that the local degeneration of the presynapse can be halted and you can slow down or prevent neurodegeneration phenotypes. And for that, we can work in flies where you can do genetic screens to understand. And this is working in collaboration with Professor Shiva Vasu's lab, where we can look at autophagosomes that are going in the neurons using neurons that contact the muscles. So these are neuromuscular junctions, and you can look at autophagy, how locally autophagy is happening. You can throw in proteins that cause aggregation, such as the ataxin-3 protein shown here. I don't know if you can squint your eyes, you may see some dark colored dots here, and those are protein aggregates that are formed. And what we show that by inducing autophagy genetically in these particular neurons, we are able to clear the protein aggregates, and these flies can now live long. Okay, this is one of the last slides that I have. Don't have time to talk about this, but mitochondria also play a very vital role. And in cells such as neurons, it turns out that the mitochondria will live as long as the neurons. Some of the cells in our body do not divide, or nor they die. They live as long as we live. And so for such cells, it's like your house in your village. Your grandparents have stayed in that. Their forefathers have stayed in that. You are going to sometimes go and visit that place. You want that place all the time clean, and you don't have If you don't have autophagy going on in this long-lived neurons, then they're going to die as well. So even the organelles health has to be kept in check. And so this is what you're looking at here are green colored mitochondria and red colored lysosomes. And at some places here, if you see carefully, you may see some yellow overlap. That's the place where mitochondria and lysosomes are overlapping. What the lysosomes are trying to do is to take away damaged mitochondria and clear it. If this process doesn't happen, that means if you have accumulation of damaged mitochondria, it's likelihood that the damaged mitochondria will have ROS production, will have protein aggregates. And now the theory is that these are the drivers for neurodegenerative diseases as we age along. So efficient autophagy in such long-lived cells is important to understand. Okay, so these are some of the problems that we are addressing. With this, I will try to acknowledge my team. It's easy for me to stand here and talk. So, but since 12 years, my students who have done all the work with the kind of different topics that I've spoken to you about. And we have a tradition that all the autophagy genes are called ATGs. So all the 6 PhD students who have graduated are called ATG 1, 2, 3, 4, 5, and 6, and several more are working on it. I showed you some of the yeast work, what Ruchika is spearheading. The salmonella work was done by Veena, and now we have Aarti, who's doing listeria to understand how listeria is different in the process. The Drosophila work is spearheaded by Anushka, and then other forms of autophagy, which I didn't have to talk about, such as cancer. Irene is working on it, and Kuku is working on the mitochondrial part. JNC is an excellent place to do research. We get good intramural support, and I've been fortunate to get some funding from many different places that are acknowledged here. This is one of the cover pages we made into a Lancet journal a couple of years back in e-biomedicine, where we show that autophagy modulators can act like vacuum cleaners, suck up the garbage, protein aggregates in the brain and clear it, and perhaps slow down or prevent neurodegeneration. With this, I'd like to thank you for your attention and look forward to taking any questions.
Thank you. Thank you, Dr. Ravi. It's a very insightful talk on autophagy. All of you might have felt the importance it is about the autophagy in our regulating our system, probably protecting the disease like cancer and during the process. I have a one or two clarifications. You mentioned either the process of accelerating the autophagy to control the cancerous cell. Sometimes you can also regulate the decreasing that due to the convenience. And during the process you mentioned about the degradation process by autophagy as a energy source. Under what circumstances autophagy is the source, the energy source?
Yes, it's a very well studied thing. In fact, the Nobel Prize work was identified by starving the cells.
Okay.
So if you don't give external source of food, cells have only two choices: either do apoptosis and die, or generate energy from somewhere within themselves. And so I think the idea is that when there is plenty of food outside, cells make more proteins, more organelles and things like that. When you don't have enough food, you have to downsize your cellular environment. And that downsizing is sort of done by autophagy, loosely speaking. So autophagy will degrade excess cellular components, superfluous organelles, and by degrading them, you're again getting amino acids, sugars, nucleotides, and things like that, which can be used for anabolic process for survival.
Okay. In continuity of that, you mentioned yes, the declining the age advances.
Yes.
Leading to that, so many neurological diseases. One of them is the Parkinson's, etc. You mentioned examples. Under any correlation between the different category of people at the particular age group, some of them are with very good autophagy rates, some of them are not. What probably the reasons? Some of the with the good habits are maintaining that. Whether what particular reasons that maintain the status quo of the autophagy in some of the people, in some of them it is declining.
So this is a very emerging question. I think a lot of study needs to be done, but it is very clear that with age, autophagy declines. But people are now doing something called intermittent fasting that most of us sometimes do for religious purposes also. But mice or most organisms that are intermittently fasted, so that for a small period of time you're fasting, that results in clear upregulation of autophagy in many tissues, but not in all tissues. So that is one natural way of boosting autophagy in your system. But I think lifestyles, including stress or associated with the unhygienic conditions and all, probably will also result in engagement with autophagy at some point of time. But whether your genes are going to help you to do better autophagy or less autophagy, only time will tell.
Whether it is possible dictate the term to do that job?
Sorry.
Dictate the gene to do that job.
Yeah, that's what we are saying. So if you have a gene that, let's say there are transcription factors like TFEB that have been identified, which can boost autophagy, right? In your system, you have more efficient TFEB functioning or some more autophagy doing proper job, that can be beneficial for you as compared to anybody else. There are single nucleotide polymorphisms. That means there are subtle changes in genes amongst all of us here. When you have external pressure such as stress or infection, then I think it's a natural selection that will happen in a population with people with better genes of autophagy will be able to sustain and fight the infections better.
I have a question. Like, are there some tags or something which says that yes, now I'm ready for autophagy or something? Because it looks like it's a general process. It's not selecting, but it's okay. It can do a mitochondria. It can remove away a chloroplast. So are there some tags which are there?
Yeah, it's a very good question. So there is general autophagy and selective autophagy. Or autophagy can pick and choose what it wants to eat, and that is dependent upon either ubiquitin dependent or ubiquitin independent processes. So if the cell has an ability to mark something that it wants the autophagy to eat, it will coat it with ubiquitin in some cases.
Similar to the ubiquitin pathway.
Yes, except that the proteasome has a smaller tunnel size. So anything that is bigger than that, autophagy we think grabs that and degrades.
And the other one was, like you said, that it usually occurs not in the older cells or something. So, but in plants, I would expect that it would be better if it occurs during the senescence process.
Yes.
So that it can remobilize all its resources back to the grain.
Absolutely. So people have shown this, that in senescent and older, whether it is yeast cells or plant cells, all the way or mammalian cells, if you increase autophagy, there are many beneficial effects. So if you make transgenics with the increased autophagy capacity, even at the during senescence, you can get a healthier plant life. Yeah.
Any questions?
Sir, I have one question. Actually, in Alzheimer's and Parkinson's, there's a aggregation of this alpha synuclein and beta amyloid.
Right.
So, and there are the different types of aggregates, So the most toxic ones are the oligomeric forms of these aggregates.
Right.
Now, my question is like, is it targets the
Extensive knowledge of agriculture, food, and drug industry with over 5 years of experience in United States. Dr. Basuraj, sir, please.
Thank you for that nice introduction. Distinguished guests and scientists and participants of this wonderful event. This is an opportunity to share my entrepreneurial journey and scientific advancements, and also the commercial, connections we have made with this industry and the customers. I think I have been given with 15 minutes plus 5 minutes of presentation. 10 plus 5, thank you for that. So I will share my technical, well, I don't want this to be a monologue, but I will rush through a few of the PPTs, what I have done as an entrepreneur in this space of biofertilizer, biostimulants, biopesticides, and PGPRs. And then I will come back to more of, maybe I'll rush through this in 5 minutes rather than 10 minutes. It should be focused on many of your interests, many of your questions. I'm going to talk about this through solutions for sustainable agriculture. All of you know, agriculture is at crossroads with many of the challenges, many of the, limitations which we are currently experiencing, be it climate change, soil health, soil fertility, crop nutrition, disease, pest, food production, residue, eating pesticides, right? All of this is, happening, and we are actually at a crossroad to design and decide how do we move the needle towards sustainability and bring better food, better nutrition, and better health. I run a couple of companies. Actually, it is called CAR, Creation, AgriApp, and Rico. I started my entrepreneurial journey in the year 2007, based out of Texas, the day I graduated, with an advice from Dr. Norman Borlaug, to get a little bit of, some advice and support. I don't want to go through all of this. This is about me. We have a clearly defined vision and mission. Our vision is pretty clear: connecting soil to soul via happy crops. Happy crops brings healthy foods, and healthy food is a basis for our healthy existence. I run very active R&D, started in Dharwad Agri University. I was recollecting with my friends, a while back. And my story was being anchored here at GKVK. And I have very active lab in Dodbalapur, running, biofertilizers, biopesticides, biostimulants, organics, alternative crop nutrition. We have few patents just to claim that we run active R&D lab. So, and another one of the important thing is the Agri app, which I am building, global agtech platform, was born out of this campus in the year 2014. I feel really connected to share, and also I have a residual or little presence here in this campus, but I would like to look into engaging more comprehensively on that. This is our R&D labs, and this is our production units. This is our capacities, presence of market, collaborations, and B2B. You all know this multi-nutrient. I'm setting a context actually for why biofertilizers, biostimulants, biologicals, biopesticides, alternatives. When we were in 60s, there was only one nutrient which was deficit: nitrogen. Today, if you look at almost all the nutrients, essential nutrients are deficit, unless, the farmers are taking care through, organics. Well, I don't need to emphasize this, but living soil versus dead soil is becoming a big debate, including, Sadhguru Jaggi Vasudev is transcontinental in, save the soil movement. But we, the scientists, we got to look at, okay, how is the life doing in the soil, how it is evolving, how we are affecting it through intense agriculture. Those are the few questions we need to look at it. integrated nutrient management, INM and conventional farming. A lot of people ask, there is a lot of research also going in this campus, not that it is not happening. The conventional farming, traditional farming, chemical farming, organic farming, natural farming, we are at a crossroad, and recently we have come across Sri Lanka's grand national experience and experiment and failure. In this current scenario, we have to go for integrated nutrient management, integrated pest management, and reduce what hurts, include what helps, keep away what doesn't really contribute. However, if we got to do a bold and a beautiful thing is precision agriculture, where we have many advantages of taking science and technology our side, especially the agri-tech part of the story, which is being built in this country and around the world. So I'm a strong believer of, agricultural science to agricultural technology, and eventually graduating to precision agriculture, where we practice sustainability and science and technology meeting the commerce. These are our few of the products. The range which I would like to emphasize is biofertilizers, biopesticides, biostimulants, and innovative products. We have many of the initiatives. Please alert me, sir, when I run to 10 minutes. The organic, we have some patents in terms of distinguishing and bringing together some of the formulations. I don't want to get into, the details of it. Biofertilizer is a wonderful thing. I worked on biological nitrogen fixation in the year 2000 to 2002 for my master's. So I got to see this wonderful biological factory in nodules of legume. That inspired me to, see how biological factories are being built by a bacteria and the legume plant. So there's a humongous opportunity, as the role of biofertilizers is enhancing, increasing, and relevance is, I keep going to US and attending these global conferences. I have seen this, friends. I have seen this trend in last 3 years or 4 years, a humongous emphasis, number one. Number two, a great amount of funding and a good amount of products. I went to this World Microbiome Conference in 2020. I see some of the companies funded in millions, hundreds of millions of dollars, not in tens. So this is a golden opportunity for us to reflect and take. By the way, our biofertilizer research is no less than anywhere in the world. I would like to, bring my understanding to the distinguished, researchers and audiences here. So, but we need to look at, okay, what we need to do so that this becomes industry relevant. Your research becomes industry relevant. Our research becomes translated into products, which customers wants it for their economic gain, and also sustainability is built into our products. If we could be able to do that, that would be a wonderful research from biotechnology, biofertilizers. The PCPRs, which was mentioned in my talk, plant growth promoters, regulators, we have a bunch of these hormones. They will help, and we can add to enhance certain physiological processes so that we are managing and sometimes controlling these processes, leading to, all that good and great. So pesticides, biofertilizers, PCPRs, biostimulants industry is very, very, very big. We need to pitch in and chip in to crop nutrition and crop promotion and growth in a big way. That would come from conferences like this. We have biofertilizers. As I said, I don't want to monologue and sell myself too much in this audience. We have fantastic product range, Gen biofertilizers, bio NPKs, liquid, solid, granules, microgranules, you name it, we have it. And I'm just waiting to evolve. I'm a big fan of 1990s when I was like sitting as a student, imagined the genetic engineering for good and great. I was, infatuated with upregulation and optimizing the nitrogen fixing genes, so that currently what we microbiologists and plant physiologists claim, 15 to 20 kg nitrogen fixation by biofertilizer should be 10x in terms of effectiveness, so that we don't need to much apply external nitrogen to the rhizosphere, where these biological factories for BNF could obviously do a miraculous job and reduce the crop requirement for nitrogen, and also help us avoid this disastrous, process of applying urea. So these are some of the relevant, the thoughts for us to look at prospectively. There is a whole lot of things happening, GMOs for India, for the world, and for what's happening in Europe and what's happening in US. Developed countries are divided for GMOs. I think I need to reiterate to my scientific colleagues here, we need to take the right side of the story. If it matters, if it is good, if it's great, we need to jump onto the GMOs. Otherwise, we need to, we should take the science basically. So biostimulants, there is a humongous market size, 5 billion. Look at that, 5.69 billion by 26. So why biostimulants? Well, for our sports people, why we give supplements, right? So is going to be our all crops, and especially specialty crops needs biostimulants. We are having all that what needed for manufacturing, research, manufacturing, marketing, and building an industry. So I wanted to present my industry experience and perspectives, and also seek your active involvement into this industry, so that we are growing this industry through research, through development, through collaborations in the industry, so that we are bringing a balanced and sustainable offerings to our customer enterprise called crop production. We have total 20 pH 50 amino acid, highly distinguished products. We sell in hundreds of thousands of metric tons of these ones. And this is again another interesting stuff we are doing, tablets for crops, basically repurposed and repacked the essentially concentrated formulation into tiny tablets. And this is good for leafy vegetables, flower crops, terrace garden, high value crops. Amazing response. Well, we are collaborating with multinational companies to come with new generation of fertilizers, which are biochemical and enriched with biostimulants and biologicals. I'm so happy to pronounce and also present, biofertilizers and biopesticide enriched water soluble fertilizers today in this August gathering. And also I'm a big preacher of the technology. I went up to many of the dignitaries and asked many of the things and trends. Now it's going to be the agri-tech times, and precision agriculture has to be brought to our farming community in an affordable, accessible, and available format. We are doing some bunch of initiatives. We have a 2 million downloaded app. I request you to explore this, because our science and tech and commerce should help sustain our farmers in terms of their enterprise, while contributing to economy and ecologically in an equal, importance manner. So I don't know, again, this thing, but that is the corner. You see the Agri app, you can download and advise me. These are digital agriculture enablers because of smartphone and data availability. And we have a bunch of drones. We're doing a full stack agriculture, agri-tech and precision agriculture. I would like to say that full stack, digitally, physically, and precision wise. This is my favorite project for sugarcane. It's called Smart Cane. Anybody farmer collaborates with us, 100 metric ton is assured. However, they have to subscribe to the digital stack, and physical POPs got to be operated. And we have live some 10 farms in Dodballapur to see and experience and touch and feel this project. I think that's all from my side in terms of biofertilizer. 10 minutes, I could not cover much, but my whole thought is to emphasize biofertilizers at need of the hour. Soil fertility revolves around 3 important aspects. All of you know, physical properties, chemical properties, and biological properties. We need to re-establish the rhizosphere with a lot of biodynamic fertilizers, biofertilizers, PZPRs, which not only does a nutritional aspect, PZPR aspects, also protects rhizosphere from, the bad organisms and bad, especially the grubs and insects. I look forward to, even I would take any comments or questions. Then I have a call for action to our researchers and potentially industry colleagues. I would do that at the end. Sir, over to you.
Good afternoon, everybody. Dr. Basuraj is entrepreneur. Still, I say in the offspring, great potential is still to come from him. Say when the industry, particularly agri-industry, whether it is agri-chemicals or medicinal sector, greatly depends on agriculture. For example, medicinal and aromatic plants and development. As far as agri-chemicals are concerned, they will fund several thousands of crores R&D. And it is good EAS have MOU with young entrepreneurs like Basuraj. We wish him to get many more such formulations, and the way in which, so truly speaking, you have patented more technologies than university did it. So we want many entrepreneurs like this, whether it is biotechnology or something else from agriculture university, should not restrict to their labs only. And in fact, biggest bane in the Indian science and technology is hardly we take up these technologies to the commercialization industry tie-ups. Just to say, even non-technologist, say petroleum dealer turned into an entrepreneur, able to launch one of the best biotech company in India, got Padma Bhushan and World Food Award. He is not biotechnologist, not BS Agri. He was managing his petrol pump. So what is the message from this? One side, Basuraj, a technologist, ordinary man, a Marwadi. So we lack that entrepreneurship, enterprising. And that is, we can see from entrepreneurs like from Basuraj, and he is nothing but B.R. Barwale of Mahiko Monsanto Biotech Limited. He was virtually a financial lender, businessman. I wish many Basuraj will emerge from university, contribute to the cause of hunger and to solve the problem of hunger in this country, and reduce the cost of production by innovative products, and increase the income of the farmers, double the farmers' income. And thank you very much for good presentation. And now it is open to the students particularly. Any questions? In fact, we have finance. You want 1,000 crores, venture capitalists are ready. Technology, you can get technology from anywhere in the world. Human resources, the best human resources are there in the world from India. What we lack is entrepreneurship. We have technology, have raw material, we have human resources, and this is the need of the hour. And this industrial interface is to be initiated by almost all universities in the country. To a certain extent, Indian Institute of Science and IITs have done it. When it comes to the agriculture university system, greatly we lack this industrial interface. And we can say from a good initiative from Dr. Basuraj, I congratulate you. And now once again, I request the students to ask some questions or some doubts in entrepreneurship and industry interface. Thank you.
A very good presentation, sir. And your products are doing very well in the market. I have a couple of questions. One is biostimulant. You told about it. How do you like differentiate biostimulant with a biofertilizer?
See, I think obviously I'll answer from regulatory standpoint, because when you take the product in the market, you need to be licensed and permitted to sell. Biofertilizers are very well defined in fertilizer control order. Your nitrogen fixers, P solubilizers, zinc, or for that matter, K mobilizers, whatever you call as per the schedule. In the nitrogen fixers, you have many more subdivisions, right? That is where the story ends for biofertilizers. Living cell, as per the CFU, performing that particular function of either fixing, mobilizing, or solubilizing. That's biofertilizer. Coming to biostimulants, it is again recently defined and put it in schedule of FCO. It should stimulate either, but non-mineral could be enriched. Okay, I can quote an example of your potassium humate, seaweed extracts, amino acids, anything innovative which you think could stimulate the crop, but you need to demonstrably have the results and proven test report from agricultural universities. That constitutes biostimulant. So there is a distinct regulatory licensing regimen for both the products.
Yeah, there are some of the company who are coming out with the microbial extract, and they quote it as a biostimulant. So they are like kind of a biofertilizer extract only. So how do you like biostim?
To answer to that, see, biofertilizers is defined very well in last 10 years by Government of India in terms of formulation and product. Biostimulant is emerging. And recently, couple of years back, the notification has come. Still, government is figuring it out with academia, research, industry, customers. How do we structure this? I'm not an authority by all means, but I can share what I have done. I'm one of the earliest to get G3 license from central government in terms of biostimulant. So based on my experience, you can go with generics, then innovate, apply, get your test report, you can include and insert your products into your G3. So yeah, I hope I answered with my limited experience. And of course, government is figuring it out, that piece of the story. Yes, please.
Very nice presentation. So your bioformulations, I hope they will be a consortia. Is it so?
See, you need to define your product. As I said, you have nitrogen fixers. When the
There
regulator
will be more than one organism. That is what I
can keep, but
yeah.
when the government labs check, you better comply the license part and the label claim part.
Fine. When new agents, bioagents get added, how do you upgrade your products?
See, R&D is fantastic. Development is also fantastic. When you put it into commercialization, we need to go and request. As I said, the regulators are always many years behind the innovators or technologists. So well, we need to go and request the government to include this in the list of FCO products, unless you need to go via the biostimulants. Otherwise, you can run this experiment as a customer demanded product. So that's where the agri-tech is coming in. You can sell on with your customer or problem specific solution product on Amazon. See, this entire distribution and the retailing via the compliance of the FCO or CIB becomes complicated. So probably I would not comment much. Again, I'm not an expert on that. Yeah, consortia are good. You can have a NPK consortia, which is permitted in FCO. If you would like to further dig in terms of commercialization, I request you to read the biofertilizer schedule in FCO. That will give you.
Madam, our legal system is so obsolete. British laws are ruling. As he rightly pointed out, it is tough job to get approvals from the government. Fortunately, in the last 5-6 years, lot of legislations were withdrawn, new legislations were brought in. Maybe I think some 3-4 days back, they are given permission to try with this says for many transgenic that plants permission was given
GMO.
4 days back. For brinjal, not given. That's only one example. Lot of virtually revolutionary legal system supporting science and technology, trade and commerce is need of the hour. And currently they are in that direction, but expectations are, as he rightly indicated, that is the legal environment that that is there in developed countries is one of the reason for many inventions being protected through trade-related intellectual property right. And USA boasts itself as the country with highest number of patents, and their legal system is one of the reason for that. And am I right,
Yes, sir.
Basaraj?
I
Thank
think
you.
that's a good point. So if there are no questions, I have 2 points, please. Sir.
I missed much of your talk. You're talking about bioagent and consortia. Are you in any way developing consortia fungus for some treatment? I mean, why I'm asking this, have you heard about agar? Agar plant? It's being introduced in Malnad area. Agar plant. It grows beautifully in 7-8 years, and it has to be injected with a consortia fungus. Many industries are doing that. I think imported one. They inject, it is supposed to stimulate the plant to produce agar. But people are complaining. It's basically a consortia fungus. When in nature, plant gets infection from that consortia and you get agar. And it's a costed plant once it is done. And you have you heard about this, or are you?
No, I haven't come across, and especially my bunch of scientists are sitting here. They run beautiful experiments in terms of designing, these consortia and biofertilizers. Number one. Number two, we don't want to get into introducing a product unless if it is government gives you a license. It becomes contravention to the regulatory ecosystem we have. So in this instance, we haven't tried that. Probably we would explore. We are doing some other thing, in terms of specific, biological action in terms of fixation, mobilization, solubilization. These are the only three things. And the fourth is the PGPR properties. Fifth is biocontrols. We have very active programs in terms of secondary metabolites, which are biologically derived, formulations which protect and build resistance for some of the bacterial, fungal, and insect pest loads. Fantastic. So if there are no further questions, I take this opportunity to thank, and I have 2 call for actions to my, student friends and researchers and industry. Please.
Hello. Sir, initial days of your startup, how you faced a problem with the farmers for your product, to convince your product?
That's wonderful. Yesterday, I think day before yesterday, I was speaking to Pusa Krishi, how to start a startup. I will share that PPT with organizers. You can share because many of the students are here. Number one. Number two, problems are plenty. I just out of the PhD, Texas A&M University, I started this creation. I could not do anything for 9 months in the US. The, bad economy, financial, problems there in US, no money. I quickly relocated to India, Bangalore, and then raised some money. I could not leave or, the startup story and leave that with many reasons. So there are plenty of problems. I can take up this explanation, or I can share that Pusa page in case if you are interested. I have answered all of this and many questions day for yesterday with that startup talk.
Yes.
You can get in touch with. The problems are plenty. You need to navigate. In case if you define a startup, go through an incubation program, and you can figure out being an agile, lean startup with that mentorship programs, right? So there are problems for sure, and there are solutions for sure. There is a luck for sure, and there is you being strong for sure. So if you could put all of this and run the program, and you need to, I typically tell my young friends, you're too young to, go with one plan. You should have a plan A, B, C, D, E. If A doesn't work, B, B doesn't work, C doesn't work, D, that is anyway dead plan. E plan is exit plan. Right?
Thank you, sir.
So that's my strength and magic. We take quick time, the lean startup stories and agile and this developmental frameworks which IT people used are secret recipes for me and my labs to quickly translate, the idea into product into commercially customer response, especially the farmer response, product performance, and the things. We sometimes take very short time. I may not be able to define because each of these products have taken their own time, no matter what I wish for. So, but this is very important question in biology and biotechnology. I know, it took 14 years. I'm in, and I've been working 20 years plus, and I fell in love with only one thing called biotechnology and genetic engineering in my undergrad. So it's taken a lifetime to do something meaningful, and we are working on genetic engineering as soon as government gives upregulating our genetic engineering for biological nitrogen fixation. This is a call for action for my research and industry colleagues. If you would like to look forward once the government opens up the door, if you have any active, interesting program to, enhance biological nitrogen fixation from 15 kg per acre to 150 kg per acre, we can avoid adding urea to rhizosphere and make urea the history. That is the power of biotechnology, which I actually imagined in my master's program, went to ICGB, whatnot. Did a little bit as a student, rebellious student. I challenged my professor, madam, I'll do this axe backs and engineering of all of this, and then realized it's not that easy. Nothing is easy, right? But we need to challenge now with CRISPR-Cas9 and all of these latest technologies and many things. These are very much possible, I feel, because we need to sell the hope to young researchers. Anything is possible. It may take your 1 year or whole lifetime. That's other part of the story. With this, 2 call for action, one for the students and student researchers to become an entrepreneur or entrepreneurial in your approach. Enterprising and entrepreneurial approach defines your new dimensions in your life, be it a researcher, professor, scientist, or industry professional, whatever you are, I suggest to become, because times in next 5-10 years are going to be altogether different. There is an end of era for jobs. There is a beginning of startup stories. How you become yourself an entrepreneur wherever you are, whether you are working for somebody, if you become an entrepreneurial person, you have 10 times opportunities. You don't need to start necessarily investing in becoming the startup owner or company owner, but becoming enterprising, as Professor emphasized. That is number one. Because I run an industry, I am hiring hundreds of MSc agree, PhD agree engineers and all. We look for enterprising ability, to run that program, project, and mission. The other was the industrial researchers and academic researchers to look at designing microbes. Now, isolation, identification, the era is good. We have some biofertilizers which work, but how do we should engineer a microbe which helps us to achieve urea mukt rhizosphere, right? We can add to the leaves and phyllosphere based on the nitrogen deficiency. With these few notes, I take this opportunity to thank organizers and all of the esteemed, distinguished, people here for giving me this opportunity and listen my talk and story. Thank you much, sir.
Thank you very much, sir. Now I request chair and co-chairpersons to hand over the memento. So we'll move to the next speaker. Dr. Narasimha Prasad, MD, Samartha Life Science, Tumkur. He's joining by online. Sir, please share your screen. Sir, we are audible. Dr. Prasad?
Yeah, I can hear you. Hello. Nagesh, ಅದು enable ಮಾಡಬೇಕು ಅನ್ಸುತ್ತೆ ನೋಡಿ ಇದು.
To enable your screen, sir, you please start share your screen and it will go ahead.
Hello.
Hello, sir, you are audible. You can go ahead.
All right. Okay. So thank you. So I think it's an honor and privilege to be part of the Silver Jubilee celebration of the Biotechnology Department. And to begin with, my apologies for not being present in person. Okay. So I would like to just share my entrepreneur journey. So through this presentation. I've been part of the pharmaceutical industry for last 20 years, of which last 15 years have been as an entrepreneur. Okay, so so my call for becoming an entrepreneur, particularly in the pharmaceutical biotechnology, was initially to look at products, which can be import substitutes, like which unfortunately is the situation even to the current day. Like there are a lot of drugs, which are imported from outside the country, and so there is plenty of scope which for these products to be produced indigenously within the country, and that's something which we can look into. And also, like in the current situation, if we look at what pandemic has thrown up, is like there's rapid strides with the pharmaceutical biotechnology has made, in terms of either the approval or the rapid release of the mRNA vaccines and DNA vaccines, and then of course the cocktail of monoclonal antibodies which have come to the market. So all this could happen, because there was an emergency requirement. And also going along with that, so if you've seen the things, like there was a huge shortage of the conventional drugs, so which were otherwise regularly maintained. So say for that matter, the anticoagulants which were required initially, and then the antibiotics subsequently. So there was a huge shortage of that, and for which the a lot of efforts were made, like government had to pressurize the pharmaceutical industry. So we here at Samarth Life Sciences, we are involved in the manufacture of some of the anticoagulants like heparins, and we have to really, put in, the efforts in terms of, one is maximizing our production, and also basically the availability of the raw material and the whole components that goes into making the drug. So that's something which was quite challenging, and I think based on that, the government has realized then, and then they have come up with schemes, so at least to reduce some of this import dependence, so that the indigenous manufacturing can be encouraged. So there are incentives given, subsidies given for the setting up of the industry, or for the development part of such drugs. So most of it which are covered are antibiotics, but there's a big scope for the other biological extracts also. So, and coming to the topic which I would like to present today is the role of these biological products and natural products, and their relevance in the current day. Because though the biotechnology has moved, but still these are products which have been conventionally made, and then at least these are there since last 30 years, and some of them could be from the last 50 years, but still their relevance is not lost. So, just to, I mean, some of these things, may appear as facts, which are obvious, but I think it calls for the attention, in the current day. So that's why, I would like to emphasize particularly on these topics today. And when you say biological products, these are products which are either live cells or products which are extracted from live organisms. So like vaccines, blood and blood components, allergenic, somatic cells. So this is by definition, these are the ones. So biologics are either they're carbohydrates, proteins, or nucleic acids, or a complex combination of these. Whereas natural products, these are products, which are either metabolites or byproducts which are derived from the living organisms. And chemically, they are classified as 5 groups, terpenoids and steroids, fatty acid derived substances and polyketides, alkaloids, non-ribosomal polypeptides, and shikimate derived compounds. And these are some of the products which are as extracts made from human or animal origin. So of which some of this are made at my company, like heparins, pancreatin, hyaluronidase. So these are made from tissue extracts of the animals, and then gonadotrophins, so which are made from human urine, and plasma-derived products, of course, which is made from human plasma. So the progress has happened in terms of converting these from their natural source or natural origin to the recombinant source. Like say this matter gonadotrophins and then hyaluronidase have been successful, whereas plasma-derived products and the others, there's still scope for the development. Similarly, I mean, this is just to give you an idea, like an industry which is set up with using the animal extracts can be it may look something like this, where this is the example of insulin being made from pig pancreas. So when it comes to natural products of microbial origin, so there are several of them, which are relevant, till date. So these are some of the products which come from fungi. Like there is compactin, lovastatin, pravacol, so caspofungin, anidula fungin. So then we have cryptocin, fomols, cyclosporins. So similarly, so we have products of bacterial origin like tigecycline, telithromycin, so ertapenem, daptomycin. So these products of microbial origin. So the requirement for this is the fermentation industry. So you need good cultures, so which can be grown or which can be multiplied to a large scale, and then you have the processes of upstream and downstream. So how do you make these produce on a on a larger scale at good yields, and then how do you purify them to meet the therapeutic standards? And I'll also look into the commercial part of it. When we say these biological products and natural products, so in terms of drugs which are approved for human use, if you see that, so more than 70% of the drugs are of these conventional biologics and natural fermentation based products. And fermentation is one thing which is crucial here. And though in the 60s and 70s, India had a good number of fermentation based industries. So gradually they declined, and so the China industry, the Chinese industry, and the import from China, so at the same time increased. So since 90s, you can see so how it has progressed, and as of now, nearly 70% of our API, that is the active pharmaceutical ingredients. So these are the substances which are required to be used in formulation. So this comes from China. And again, this is the import data. So 2021 of the crucial API, so which is coming from China. So if we see here, so most of it is either 100% coming from China, or nearly 70 to 80% of it is coming from China. So one is in terms of the quantity that comes, and the value terms, the foreign exchange, so that is transacted with China. So it's a big dependence there, on the Chinese supply. So just to give you a number on penicillin G, so which is the most crucial API, which is also used as the starting material for some of the other antibiotics also. Nearly 6,000 tons of penicillin G is imported from China. So when we look at this, so what led to the decline, and so how is it that China is more competitive than us, in terms of manufacturing, and just to look at where we can improve. So if you look at per unit cost, if for something, say, assume like something costs you 100 rupees here to manufacture, so it would cost something like 81 rupees in China. So there is that difference of 20% over the cost. That's where they gain over us. So for that, so one is, so they get cheaper raw materials, so they are 25 to 30% cheaper than us. So, and then electricity, so we are 20% expensive than China. So only thing which is on our side is the labor. So we are like 1.8 times lower than China. And then the other costs, of manufacturing, the packaging, and then most importantly, the scale, the scale at which the Chinese companies operate to what we operate. So there's a big difference there. So this overall contributes, to this 20% difference, and that's where we have become less competitive as compared to the Chinese companies. So what this requires is, I would just like to focus on the fermentation, and with respect to, if you just look at only the antibiotics part of it, so, the need of the hour is not much of, whole scale innovation or a large scale innovation. So which to bring out, say, a new organism to produce a new antibiotic. So that's something, which can happen. And, if you look at the lead time of developing a drug, typically it takes at least 10 years, for you to qualify this as a drug for human use. Whereas what is required is incremental innovation, so we have strains of microbes, so which are used to produce these drugs, but we need to make them better yielding strains. Similarly, we need to bring down our cost of culturing these microbes, so use of alternate or frugal media, so that can bring down the cost of the fermentation. And then the productivities, improving the productivity overall, the upstream and downstream process of the fermentation needs to be improved, so that so that contributes to a component there. So of which, the if we, when I just looked at the data of, how better the strains that are used in the Chinese industries would be as compared to what are used in the Indian industry, the difference is only about 3 to 5%. I think that's something, which comes under this incremental innovation. So if we can catch up to them, so by this 3 to 5%, and then, with the other aids like the incentives that are being provided by the government, and then, we need to improve our scale, make it more productive. So I'm sure, we should be able to catch up with them. And this is one area where, so we can look at immediately solving the issues that we have with respect to the imports that are coming from China. And I believe the industry academia support or industry academia collaboration is required here, so that so can work together to match this difference which is there, so that, so the import dependence, which I said initially was one of the areas, which I could look at it when I started my career as an entrepreneur. So that's something, which can always be looked into. Yeah. So yeah, with this, I would like to end my presentation. So before I complete, so I would like to remember here Mr. Sudarshan. So I'm from the biochemistry department, the erstwhile biochemistry department, and Sudarshan sir was one of our guiding force then, and who encouraged people like me to, find a foothold in the industry. So we'll be always thankful for him, and I think it's time to remember him. Yeah, and thank you. And yeah, I'm open for any of the questions. So particularly the graduating students or students may have. So if you're looking for entrepreneurial journey, so I would like to say here, like, there is a big opportunity waiting for you in the pharmaceutical sector, diagnostic sector. Because as I said, so we can't just look into only the new age biopharmaceuticals, because development of a biopharmaceutical or a new biological, so it needs a huge amount of capex and the operating costs, and obviously you'll have to be patient with time there. But there are other areas, which need to be tapped, like many of these extracts which comes into diagnostic industry, they're all coming from either Europe or China. And obviously, many of these APIs which I listed there, so they're also coming from Europe and China. So this is something, which can be looked into for those of you, who, so if you just check the import data, and wherever, we are depending on other countries for these products for our Indian market supply. So one of them can be tapped into it, so it can be capitalized. Yeah, thank you.
Maybe there may be some difficulty in asking questions to him. Maybe through that laptop you need to ask. Okay. My fellow scientists and students, pharmaceutical industry is the biggest industry in the world, even much more than petroleum industry in the world. And if you look at the lobby in the pharmaceutical industry, biggest lobby starting from America. Incidentally, India is in the forefront in the bulk drugs. Last week, Government of India sanctioned bulk drug parks to 3 states, one to Himachal Pradesh, another to Gujarat, and one more to Andhra Pradesh in an area of 5,000 acres. So it is golden opportunity to the startups to try to establish your pharmaceutical companies, particularly agro-based pharmaceutical products. You can try, and Government of India also encourages for the startups. But 90% of the startups are big failure. 90% of the startups fail within 2 years. So keeping this in view, the best example you can quote how our agriculture graduates can perform, particularly in the pharmaceutical industry, is Krishna Illa alumni from this university, who was sitting like you, maybe in this auditorium 2 decades back. Now his name is known across the world. So it is right time persons like Prasad entered into this pharmaceutical industry, particularly agro-based pharmaceutical, and say, for example, Dr. Reddy Lab, Arvindo, Natural Remedies, Ranbaxy. These are the companies in the pharmaceutical involved in bulk drugs. So this big 3 bulk drug parks to be established shortly may open up avenues to the youngsters like you, and funding is no issue. Atal incubation centers are there across the country, and particularly for medicinal and aromatic plants, it is there in Gujarat, Directorate of Medicinal and Aromatic Plants. MediHub is there. You can try. With this remarks, any questions put to Dr. Prasad?
If there is no queries, I would like to thank to the Dr. Prasad, sir. Thank you.
Thank you. Thank you very much.
So we come to the end of the session.